अगर होगी वो करो oh sir we'll start with the technical program. The true meaning of life is to plant trees under whose shade we always search for. Plants put down deep roots not only into the soil, but into our whole being. Until we dig a hole, you plant a tree, you water it and make it survive, you haven't done anything in your life. Without plants, there is no food. Without soil, there is no life. With this, managing and conserving both plant and soil is our utmost priority in our life. So, we'll start today's technical program with a presidential lecture on green strategies and innovation for plant and soil health management. President IPS, CSIR, Lucknow, Uttar Pradesh. Professor Rakesh Pandey is an emeritus professor of CSIR, scientist, CSIR Central Institute of Medical and Aromatic Plants, Lucknow, and more than 39 years of research experience in the field of medicinal and aromatic plants and microbiology, genetology, neurobiology, with a special reference to the development of agrotechnologies for some of the important maps, and achieved a novel breakthrough in the field of congenital disease and so on. And he has published more than 164 publication and 18 review articles and 33 book chapters. And he has authored more than 7 books and a US patent of 4. And he has been awarded with a DAD Fellow and a Senior DAD Fellow and the Visiting Scientist, University of Pittsburgh, USA, and a Golden Peacock Award and a Professor V. White Memorial Award, Udaipur, CSIR Technology Award, Outstanding Scientist Award, and Mishra Medal, and a Young Achiever Award, and so on. And he has been a fellow of National Academy of Agriculture Sciences, Phytopathological Society of India, and a fellow Nematological Society of India, and a fellow Indian Botanical Survey, and a fellow Indian Society of Mycology and Plant Pathology, and a fellow Indian Academy of Horticulture Sciences. And his research area being developmental biology, neurobiology, nematology, phytology, and agriculture microbiology. I welcome you for the session, sir. It's our pleasure having you here. And today's technical program is chaired by Professor Apparao Podle. Dr. Apparao Podle is a senior professor, and he was a vice chancellor of University of Hyderabad and honorary chair professor in biotechnology, Asia University, Changchun, Taiwan, and a president, Association of Microbiologists of India, and a vice president, Indian Society for Chitin and Chitosan. And his area of research are PGPR, host resident, and plant microbiome. And he has given a major significant contribution in the field of PGPR, host resident, and plant microbiome. And he has been awarded with many awards and honors, like a Millennium Plaque of Honor from the Indian Society of Indian Science Congress Association, ISCA, and K.C. Mehta Award in the Crop Protection from the National Academy of Sciences, and the Rangaswamy Award in the Agricultural Microbiology from the Association of Microbiologists of India, Lifetime Achievement Award in Biotechnology by Association of Biotechnology and Pharmacy, Best Faculty Award and a Research Award, Indian Society for Chitin and Chitosan, A.C. Dubey Outstanding Young Scientist Award from the Indian Society of Microbiology and Plant Pathology, and the Rajiv Goel Young Scientist Award in the Agriculture Science from the Goel Foundation. He has also been awarded as a fellow in Indian National Science Academy and Indian Academy of Sciences, and the fellow in the National Academy of Sciences, and so on, to his credits. I welcome you as a chair for this session, sir. And today's session is co-chaired by Dr. R. Sridhar, former Vice Chancellor. His area of research being
Professor K. Sridhar from Bangalore University.
Well,
Professor K. Sridhar.
ಇದಲ್ವಾ
from Bangalore University ಅಂತ ಕರಿ ಅಷ್ಟೇ ಸಾಕು.
Okay. Today's session is co-chaired by Professor K. Sridhar, Mangalore University, Mangalore, Karnataka. I welcome you for the session, sir. And the rapporteurs of today's session is India, and Dr. D. Parma, AICRIP, Gangavati, Karnataka, India. I will welcome your work for the sessions. Presidential lecture by Dr. Rakesh Pandey, President IPS, CSIA, CIMAP, Lucknow, on green strategies and innovation plant and soil health management. So what do you say?
Honorable Chairman, Dr. Odle, Co-Chairman Dr. Sridhar, Reporter, Professor Chahal, Dr. Dubey, Dr. Kushal Raj, Professor Chakravarti, Dr. Mandal, and Dr. Kholia, and my dear students. Now today I'm going to talk, even it's not a lecture, you can think that we are going to discuss what green strategy we can make or what new innovation we can make for making plant and soil health. Because plant and soil health, that is very important for any research. Plant and soil health play a very important role. How we can? Now, it will be injustice if I will not talk about the medicinal aromatic plants. I got 40 years on different medicinal aromatic plants, and we have developed several technologies. And these medicinal aromatic plants, now farmers are cultivating in their field. Better means even for 6-month crop, farmers are getting 1.5 lakhs rupees per hectare. So this is one of the best commercial crop which is fit in our other cropping system. So these are the few crop, like we are having ashwagandha. Here is the ashwagandha. We make ashwagandha wrist. Then opium poppy, we are making, our institute is making seeds opium. We can produce, instead of alkaloid, morphine and other, we can produce more seed. Then we are having Ralphia serpentina, that is major source of the serpentine that used for the depression drug development. Then we are having Artemisia annua, that is artemisinin active constituent that used for the malarial fever. Then we are having Isabgol, that is Plantago ovata, and then Curcuma. And we are having very good Pitambar, that variety released by our Honorable Prime Minister, that is having very good amount of curcumin. That is the highest amount of curcumin till now reported. Then we are having matricaria, then some chlorophytum, aloe vera, calmate, that is very important. During the COVID-19, we have made the immune booster activity. We make one herbal product by using the Endogaphis paniculata, that is calmate. Then we are having the satavar. This is very important. Asparagus racemosus, then Coleus porciculi, that is cardiotonic we make from the Coleus. Then we are having mentha. Then different type of mints. I'm not going deep. It's not working. Then osimum, and it will work here. Okay, then we are having aromatic grasses, and especially if you talk about aromatic grasses, this is the khas. This one is the khas, and this you can grow at 9.5 pH. There is rare crop you will get when you are growing at the pH 9.5. If there is a flood, so these some of the aromatic grasses like khas, Vetiveria jejunioides, that you can grow and farmer can get more than 1.1 lakhs rupees in 9 months. Earlier, khas was growing for 2-year, 3-year crop, but our institute has developed such a variety. We can have a khas oil in 9 months, and farmer can get very good income from the usur and other type of the soil. And these medicinematic plants, whatever product we are having, then we are having very good product. These are all the herbal product made in our institute. And why I am showing this product? Because when we are talking diseases, we have to avoid the pesticide. That is the reason I am showing this product, which is made from the medicinal and aromatic plant. Now the crop losses, you see about the virus, phytoplasma, bacteria, fungi, and nematode a lot. I'm not going to tell you that it is 10%, 60%. Sometimes whole crop is going to finish. You will get more than 70, 80% loss. It depends on the crop and pathogen. So how to manage these pathogen? The best way is that we have to make the soil healthy. Before coming on the challenges, I would like to discuss about the soil health. When we are talking about the soil health, there are 3 types. Chemical, it is not working. There is a chemical, physical, and biological. When we are talking about the soil health chemical, then we have to go for micro and macronutrient in the soil. When we are going to talk about the physical, then we have to go the water holding capacity, types of soil texture. And when we are talking the biological health of the soil, we have to see how much useful microbes are there. So that is very important. And being a plant pathologist, we are having major challenges. These 4 challenges. Number 1 is that to promote the sustainable agriculture and ensure food and nutraceutical security. Not only food, nowadays people are talking about nutraceuticals. So what about nutrition is there? Because there is a lot of problem when there is a nutraceutical scarcity is there. So we have to also need a nutraceutical society. And to protect the soil and plant health from biotic and abiotic stresses, generally we are talking even about the biotic stresses, we will discuss a lot. To maintain the green and clean environment, this is very important. And when we are using any chemical pesticide, either it is fungicide, nematicide, we have to take the care of the environment health. Why I am taking environment health? Because whatever pesticide we are taking, there is a problem of neurodegenerative diseases. When people are using more pesticide, even farmers are not aware, but you see those area people are having more Alzheimer and Parkinson. Why we are having Alzheimer and Parkinson? Because those farmers, they are not aware how to use these pesticide, how to use these nematicide. When it go in their system, it direct damage their neuron. And when neuron damage is there, the person is having at old age the Alzheimer and Parkinson. So that we have to avoid. That we have to take maximum care, because last week I was in Bangalore in one of the company lecture. Then I asked those people, give me the tox data. What is your toxicological data? Because they were developing one of the product to manage the nematode. I asked them, give me your toxicological data. And that product came in the market. And I'm telling you, they were not able to show me the toxicological data. And that product is available in the market. Then I told them, this product is going to damage the neuron of the farmer. They will face the neurodegenerative diseases. Who will be responsible? Then they told, sir, I am taking your mobile number and I'm taking your mail ID. I will send you the tox data. More than 10 days over, they have not sent me the tox data. So we have to take more care. When new product is coming in the market, coming in the field, in the farmer field, where is the toxicological data? And toxicological data should be real. It should not be taken through third person. Like I know so many people in Delhi. Even they have contacted me when I was working on trichoderma. They contacted me, sir, I can give you the tox data. How you can give the tox data when I'm working on trichoderma? I'm working on my microbes. How you can give me a tox data? So such type of things is going on. Be plant pathologist. This is our responsibility to take care of these things. Because our major peoples are farmers, society, they should not suffer. This is our responsibility. This is responsibility of the IPS. If some new pesticide, new chemical is coming to manage the phytopathogen, we have to take care of the toxicological data. Then, like European Commission, they have a meeting on 22nd June 2022, and they are planning that they will reduce more than 50% major pesticide in their country. Hope they will reduce. And they will ban also. But I don't think in India how we will ban. Even some chemical are banned officially, but they are available in the market. They are available. I'm telling you the example of the furadon. When people were taking 2 kg of furadon, it is good for the nematode management. You will not believe when you take 5 kg, it is not working. You are talking 2 kg, there is 5 kg, it is not working. Because that is not a good one, because it is selling in the market, local market, people are selling in the small city, but that is not active. So such type of examples are available and farmers are not aware. And the main challenges is that how to increase the crop production through crop protection. For example, we are having post-harvest losses. Even losses caused by pathogen and pest, if we can decrease it, even we can feed our population another 50 years. Because whatever losses, post-harvest losses and other crop losses are there, even if you maintain, if you manage the pathogen, we can feed our country another 50 years without any problem. The thing is that we are not able to manage them. A lot of post-harvest losses are going on. Population is increasing, no need to tell about this, but how it is possible? It is duty of the phytopathologist, plant pathologist, how to manage the disease. Now, the question is that how it looks like in 2030 if damage is continuing. Even though we have to think about beyond 2030 or beyond 2050, even beyond COVID. In one way, the pesticide growth is increasing. You see, 78.16, 2021. 2022, they have predicted $85.11 billion. And now they predicted in 2026, $105 billion. So their growth is increasing. We have to increase the biocontrol or microbial management of the pest and pathogen. Otherwise, these pesticides, they are increasing, and what will be the future? We have to think over that. These methods are available. Biological, even consortia is one of the best. During my last 40 years of experiment, I'm telling you the consortia is one of the best way how to manage the diseases. One of the example I'm telling you that with the chemist, we have isolated one calicarpinine, one of the molecule from Calicarpa macrophylla. And when we mix this molecule with the trichoderma and some of the bacillus PGPR, we have got very good plant growth. Even disease suppression was also there. In practical, I have got that consortia is working better than any other way. Even nowadays, people are working on the gene editing, Cas9 technology. They are developing some new plant. They are resistant to pest and pathogen, but sometimes they become susceptible. Even if you see the growth of the biological and chemical, hope after 2050, the biological will grow. It will surpass the chemical pesticide or chemical fungicide or chemical nematicide, as it has been predicted now. And modern biotechnology, they are playing major role for creation of the healthy environment, for soil and plant health. This is a story of the gene technology, how people have developed a resistant variety against the pests and pathogens, especially viruses. For example, transgenic plant disease resistance against microbial, it has been demonstrated in 1986. But when they develop a new papaya against PRV in 1998, its durability was only 15 years. Again, it becomes susceptible. So there we have to think when we are taking the use of the molecular biology or biotechnology, what is the durability of the resistance? That we have to take care. Then these are the different examples are available, how the biotechnology play major role for the economy of the country or other countries also. I'm not going to develop. Now, different omics technology are available. And now people are utilizing these omics technology for developing a plant resistance against pest and pathogen. How we can utilize it against the pathogen and how a plant pathologist can work. I think Dr. Apparao is one of the best man in our country who has done a lot on this omics technology. Then when we are talking about the microbiome, it is very important. For example, endosphere. Generally, very less people are working on endosphere. But endosphere also play a major role for enhancing the plant secondary metabolite. Why I'm talking plant secondary metabolite? Because I'm working on medicinal plants, and my major role is that to increase the plant secondary metabolite and decrease the plant diseases. So that is my major part. So how these microbiome playing major role, we have to think, and we have to increase the useful microbes in the soil. And that will be very useful. Like here, one of the best example you can see when we are using useful microbiome, how the plant yield is increasing. And they are affecting on different parameters. I'm not talking one parameter. This figure you can see different parameter is affected by the soil microbiome. So if we are having good microbes, then it is working very well against the diseases. Then if you talk good, bad, ugly, then bad and ugly, how we can avoid it? By using the good microbiome. So here we have to take the indoor rhizosphere and rhizospheric microbes. Both we will take, and then we can increase the yield of the crop and develop resistance against the pest and pathogen. Here one of the best example, I am showing that how the plant develop a immunity, either it is ISR, either it is SAR, by using the different microbes in the plant. Nanotechnology, now growing area of the nanotechnology. People are using nanofertilizer, nanopesticide, nanofungicide, nanonematicide, and it is working. But we have to think these nano, they are not going to enter in our food chain. That we have to take care, because if nano is coming in our food chain, then it is going to have a great impact on our health. So that we have to take care when we are working on the different type of the nanoparticle. And even ketosane, Dr. Podele is working, Apparao is working on ketosane. He know better how people are using this nano ketosane, and they are mixing these ketosane with the different molecules, plant molecules, and they are making that anti-biofilm activity, because whatever the pathogenic organism are there, they are making a biofilm. Biofilm means then our useful microbes cannot enter. So until you are not going to break the biofilm, whatever technology, whatever things you are developing, whatever microbes you are developing, it will not work. So that is major part is that how to break the biofilm. Then our molecule or our things will work. So these are playing a major role, and that we have to take care. And then nanopesticide, already I have discussed nanofertilizer, no need to discuss here. And I want to show you some of my own work, because what I did, what my group has done. Here, one of the example I'm telling you that we have taken trichoderma and some of the microbes. And what we did, we have developed, you see, lignification against the nematode. So when we are developing lignification in the root, nematode cannot enter inside the root. So we are restricting nematode in the soil, they cannot enter in the root. So here we manage the nematode problem by using the microbes, by increasing the lignification and increasing the plant secondary metabolite, bidhanlate, bidafrin A. That is the major part of our research. Then here, another point is that we are working on aromatic plant. Aromatic plant yield only oil can increase when we are increasing the number of the trichomes. Because trichomes are main part, they make the oil gland. So here when we have used the microbe, they increases the number of the trichomes. And when trichomes are increasing, then we are getting very good maximum oil glands. So oil yield is increasing. So here we were targeted in aromatic crops, how the microbes can increase the number of trichome in the plant leaf. That is, and this work we have done and published it. Then another is Bacopa moneri. This is very important plant for the memory tonic. And bacoside is the major active molecule. And we have used trichoderma and Bacillus subtilis, and we have increased the yield of the bacoside. Because our target is there to increase the plant secondary metabolite, not only increase the plant yield. If you are increasing the plant yield and alkaloid yield is zero, there is no value. So ultimately, we have to go to the target molecule or target alkaloid. When we are increasing the yield of target alkaloid, then only it is useful. Otherwise, there is no useful if we are making more herbage or something like that. And then we have used some of the Chitinophilus and Streptomyces against the root knot nematode. You can see here the lower figure. In control and nematode is there, you see whole root is damaged. But when we have used microbes, we got the less damage in the root system. So how we use the microbes to manage the nematode problem in the plant. And not only I am showing you the practical, we have developed a technology also. And that technology has gone to the farmer field. It is not only I am publishing an impact factor journal and showing you. This technology has gone to the farmer field. So that is the beauty of our science. Because in CSIR, if your technology is not going to farmer field, it is very difficult for you to get a promotion. For scientists now, it becomes very mandatory. So we have to work for the society, for the farmer, if we want to grow our career. That is very important now. And some of the picture I am showing how these different microbes manage the reactive oxygen species. You can see in the leaf, then in the root, you can see how we are increasing this thing, how we are managing the nematode problem. And then we have taken the picomolar solution of some of the molecule, and we have increased the microbial population in the root system. Means in the root, we have got more colonization when we have taken the picomolar solution of the lutein. That paper we published in +1. And then another work we did here, hexavalent chromium. Because in the nearby Lucknow, there is Unnao and Kanpur district. A lot of the plants are affected by the hexavalent chromium. And these hexavalent chromium not only affecting the plant, they are carcinogenic also. So what happened, we have isolated some of the Bacillus megatherium species, later on we identified it, and that make hexavalent chromium to trivalent chromium, because trivalent chromium is non-carcinogenic. So that microbes not only remediating the hexavalent chromium to trivalent chromium, it is also increasing the oil yield and decreasing the nematode population. So these are the different parameters I observe. So I'm not going in deep because time is very less. And another thing, this is our technology, how we transfer this trichoderma technology, what patent we are having for patent. That technology has gone to farmer field. And if any technology is going to increase the yield of the farmer crops, then it is good. For example, if I am working, I am becoming a trichoderma man. If farmer is affected, farmer is getting yield increase, getting more money, more revenue, then it is useful. Otherwise, there is no use of any technology, just publishing a paper. So here, how we use the sector, then minister and governor, they all visited our whatever product we develop in our lab. But the most important thing that I'm happy because I'm seeing happiness on the face of the kids. And at least the farmers who have adopted our technology, they are having very good revenues, and they are making their pakka houses, and they are making even from ox, they have purchased some tractor and other things. So now they are working very well when they adopted our technology. And whatever I did, because I have not shown my all work, only few slides I have shown you. These credit goes to my student, not to me. I'm not responsible. Whatever I did, whatever I published, these all credit goes to my student because they are the major pillar of my science. What science I did in 40 years, these all credit goes to my student. So thank you very much. And this is my herbal garden, Mano. We have made a garden that every medicinal plant should be in the different part of the body. So at least a student can identify and see the use of the plant. If you want any query, I will be happy to answer. Any query?
I'm sure all of us enjoyed the presidential address delivered by Dr. Rakesh Pandey. He has brought in his rich experience of working with medicinal plants and development of different products. Sir, as a customary, I believe presidential address will not be open for questions. So on behalf of the organizers, on behalf of our Indian Phytopathological Society, I thank you for your wonderful talk. And as a president, he has given us a cushion. He has shown the way to save some time. He has reduced his lecture time and saved a little bit of time for the session and for the forthcoming sessions also. Thank you so much, Professor Rakesh Pandey. Now, another important plenary speaker, none other than Professor K.V. Subbarao. Professor Subbarao is a son of the soil, graduated from this university, obtained his master's in 78, and then briefly he worked with ICRISAT for about 5-6 years before he went on to do his PhD at Louisiana State University in the United States, and which he graduated in 1989, for which he got a very prestigious award, the Edgerton Award, C.W. Edgerton Award for Outstanding Performance as a graduate student at Louisiana State University in 1988. And to mention very little about Professor Subbarao, he is currently a professor at University of California, Davis, and he was also associate professor there. And most of us would have seen him on the prestigious journal that many of us would look at, the Phytopathology. He has been associated with the journal for more than a decade in different capacities, including editor-in-chief. That's a prestigious honor that such kind of a, and he is an elected fellow of the American Phytopathological Society. With all these distinctions, he has agreed to be with us to deliver his plenary talk, and it is my pleasant duty to invite him to share his experience on comparative genomics, particularly on Verticillium dahliae, where there is very little work on Verticillium dahliae, and he is going to speak to us on the comparative genomics of defoliating and non-defoliating strains of Verticillium, and how the horizontal gene transfer from from another wilt-causing organism would have been responsible for all that. And with this little of introduction, I invite Professor K.V. Subbarao for his plenary talk. Professor Subbarao.
Thank you so much, Dr. Padile, for that kind introduction. My heart swells with pride today because I learned the ABCs of plant pathology right in this very campus. And it also infused in me the power of observation, whether it is under the microscope or in a research field, or the general changes in the society that directly affect plant diseases, that those experiences went on to shape my career. And I'm very happy to be with you and present this talk. I know you're all hungry, We are already 2 hours late. I won't take too long, but I want to just give you a glimpse of what is possible. These are exciting times to be in science, and especially in plant biology, because of the technological changes that are available now. The kind of problems that you can solve are simply limitless. And I'll give you one example of such a problem that had existed for over 70 years. And using genomics, we were able to resolve those issues. And as you may have heard my talks in previous APS meetings, I have had a long-standing interest in wilt-causing pathogens, especially verticillium wilt caused by Verticillium dahliae. Pointer work kaada? Work kaada. Paragila. Okay. So Verticillium wilt is caused by, it's a small genus, Verticillium. It has 10 different species. And Verticillium dahliae is the most important one in the country. So Verticillium dahliae is the most important pathogen. It's a strictly soil-borne pathogen. It produces long-standing survival structures called microsclerotia that remain in the soil viable for up to 10 years. And once it is infested, you have extremely long-term problem in your fields. It causes the vascular wilt. It invades the xylem tissue, causing the wilt disease. And it also has a very broad host range. Conservatively estimated, it infects up to 250 host species, many of them including economically important crops. And it is an entirely asexual pathogen. No one has observed sexual cycle either in the laboratory or in the field. So it is a clonal pathogen. On the right-hand side, you see some of the crops that are affected by the disease. And the plants nearing harvest will simply collapse with the wilt because the xylem is completely plugged up. So wilt diseases in general have 3 different phases. The first phase is at the root zone. That is the initial combat zone. And in the xylem tissue, that's in the middle. And the ultimate final victory of the pathogen over the plant happens in the leaves by way of this one. And different components of the pathogen actually played different roles at each of these phases that are shown in the different colors on the left-hand side. And the entire array of enzymes and toxins that are produced by the pathogen, we call them secretome. The different components of the secretome play different roles in each of these phases. So we published a review just last year in Biological Reviews. And if more of you is interested in studying the details of this one, you can refer to that one. And I told you it is a pathogen of upwards of 250 crops. And in all of those crops, the normal symptom that you observe is wilting, except in 3 crops that are represented here, cotton, okra, and also olives. The wilting is combined with defoliation, premature defoliation of the plants. Certain strains of this pathogen cause complete defoliation. And on the left-hand side, you see a branch of an olive tree, 2-year-old olive tree that is already deceased. By the end of the 2nd year, it is completely denuded. And by the end of the 3rd year, the plant is completely dead. If you cut open the plant, then you see the characteristic vascular discoloration. And in the cotton, exactly same thing happens. Just when the plant begins to flower, it sheds all of the leaves when it is infected by certain strains of Verticillium dahliae. So defoliation has many causes. We have learned that it is a natural process of growth and development that is mediated by these 2 phytohormones, abscisic acid and ethylene. And certain phytotoxic compounds also cause defoliation. In fact, the 2 compounds that are within the parentheses there are actively used on cotton to defoliate prior to machine harvest because they don't want the lint to have too much of a foliar debris. So deliberately, the cotton crop is defoliated using these two. And we also know there are plenty of abiotic stresses that cause defoliation. And defoliation can also be caused by both pathogens and pests. So the phenomenon of defoliation was first discovered in California, in fact. There was a USDA scientist at UC Davis that discovered this phenomenon on cotton, and he characterized the strains that cause defoliation and non-defoliation and called them as such, defoliating and non-defoliating. And since then, there have been over 100 papers on this very topic without completely addressing why defoliation happens. And many groups had tried and failed. And it is only, it probably waited for us to resolve this issue. And I'll share that story with you. So previously, we had used a genomic, comparative genomics approach to study the differences between race 1 and race 2 of Verticillium dahliae, where we obtained the complete genome of these two and compared the two genomes. And in race 1 isolates, about that band that you see, the 50 kb region, was only present in the race 1 strains, and it was completely absent in the race 2 strains. And we went on to characterize a race 1 effector from this region that was published in PNAS in 2011. And we used a similar approach in this study also, wherein we selected a very virulent strain, VD991, that is defoliating from cotton, that is not only virulent on cotton, but is also moderately virulent on tomato, but it is completely avirulent on lettuce. And similarly, we also selected a strain from lettuce that is non-defoliating, and also the strain from tomato that was also non-defoliating. And as you can see here, only the defoliating strain causes defoliation, and none of the others defoliate the cotton plants. And only cotton is infected by all 3 strains, and lettuce is not is infected only by the by the lettuce strain. So we obtained the complete genome. These are 8 chromosomes. There are 8 chromosomes in Verticillium dahliae. And on chromosome 3 and 4, there are these specific regions that are called lineage-specific regions that are very rich in transposons that confer some of the specific characteristics to Verticillium dahliae. So when we compared those two in GSLR2, that is shown in the red box, that was completely different in the defoliation strain than the other 2 strains. And we took that area to study further. So we obtained the complete genome to ensure that it was not specific to VD991, the strain that we selected. We compared a whole population of the defoliating strain. And in all of those strains, you see the presence of that 109 kb region that is absent in the non-defoliating. And we will find out where this region came from. How did Verticillium dahliae defoliating strain acquire this region from? So we did a phylogenetic analysis comparing it to several species of Fusarium. And you notice that small box that is shown in green, that was complete, that had a perfect match with Fusarium oxysporum vasinfectum, which is also a wilt-causing pathogen on cotton. So there was actually a horizontal gene transfer from this, and it can easily happen because they both share the same niche in the soil as well as in the plant xylem. So we knew that it acquired, once it acquired, there were some deletions and additions that conferred the cotton strain of Verticillium dahliae the specific defoliation ability. So we then went on to characterize functionally how this region actually causes defoliation. And as you notice here, when you delete this GSLR2 that is in the cotton strain, the defoliating strain becomes the non-defoliating strain on cotton, but it has no effect on the other 2 hosts that we included in this study. So when you transfer that GSLR2 to either the tomato strain or the lettuce strain, it actually increases the virulence of those two strains on cotton, but it doesn't alter the virulence on either their own respective hosts. So this region we knew is the one that causes defoliation. So the next question is, how does it cause defoliation? So we then went on to, this is once again on lettuce and tomato. There was absolutely no effect of transferring this on the virulence of these two strains on their respective hosts. So we knew that horizontal gene transfer of this LSR region is what conferred this defoliation ability to Verticillium dahliae from cotton. And it is probably one of the mechanisms by which asexual pathogens develop virulence mechanisms also. So there are 7 different genes within this GSLR2 region. So named VDDF1 through 7, and we individually knocked out all of these, and then we assayed their virulence. As you can see here from defoliation strains of gene 1, 2, 3, and 4 did not have a phenotype. In other words, they still remained as a defoliation strain, even though they lacked the corresponding gene. However, when you delete genes 5 and 6 in this region, then the defoliation strain becomes the non-defoliation strain. So it is these 2 genes that are very, very important in conferring the defoliation. So you notice that gene 7 also did not have a phenotype here, but it plays a very significant role indirectly, and I'll come to that in a couple of slides. So the question then is, since it acquired this region from Fusarium, whether the GSLR2 from Fusarium or these 2 genes, 5 and 6 from Fusarium, are able to cause defoliation. And as I told you before, it does not cause defoliation simply because these 2 genes have an ineffective expression. Both need to be expressed at the same level at the same time, whereas in Fusarium, it does not happen. And because of that one, defoliation does not occur with Fusarium oxysporum vasinfectum. So then we wanted to compare the results on cotton with also olives, just to ensure that what we are observing on cotton is not limited to cotton. So we repeated those studies on olives with identical results. So what is special? How do these 2 genes cause defoliation? What do they encode? In fact, both VDDF5 and 6 are in the PKS cluster. So they encode that compound N-acyl ethanolamine, NAE, commonly called. And VDDF7, I told you, it did not have a phenotype. What it does is it encodes an enzyme that feeds more of NAE into the system itself. So that's how it plays a very significant role. So there are a number of different NAE types, and this is the flow chart for how the enzyme that VDDF7 encodes produces more NAE to feed into the system itself. There are different types of NAE, and they have various functions in the plant systems. And we wanted to find out which of these NAEs are produced by Verticillium dahliae and which actually cause the defoliation. So Verticillium dahliae produces 3 different NAE types: NAE16, NAE14, and NAE12. And on the right-hand side, you see the amount of NAE16 that is produced by both the defoliating strain and non-defoliating strain. The variation within the strains of individual group were much larger than the differences between the groups themselves. So obviously, NAE16 was not the candidate that explains the difference in the 2 strains types. And NAE14 is produced more by the non-defoliating strain. And obviously, this was not the candidate that causes defoliation either. So then we settled on NAE12 and did functional analysis once again. And as you can see here in the when you in the strains where genes 5 and 6 of this cluster are deleted, their ability to produce this NAE is severely restricted, whereas the wild type strain produces copious amounts of NAE. And if you complement those two genes in a strain, then it restores the ability of that strain to produce NAE12. So then if you put these two genes in a non-defoliating strain, whether that is able to produce this NAE12. So that is the last strain that says VDG78. That is a non-defoliating strain into which we added these two genes, 5 and 6, that is able to produce NAE12 once again. So the question then is, if you know that NAE is the chemical that causes defoliation, whether you can reproduce the defoliation symptom in the absence of the pathogen, or if you inoculate the plant with a non-defoliating strain, if you supply NAE12, does it also defoliate? That is the experiment that we did here in the bottom that you see, where we inoculated the plant with a non-defoliating strain, and then 1 week later, we supplied NAE12 through soil, and it immediately was able to defoliate the plants. And if you actually simply supply NAE12 to the cotton plant, that also causes defoliation, but it takes much longer. In the presence of the pathogen, the defoliation happens much quickly, but NAE12 alone is also able to cause defoliation, but the whole symptom onset is delayed. And we hesitated to do this experiment, and we submitted the manuscript to New Phytologist as it is, but the reviewers and the editors insisted on us doing this experiment. The reason we had not done that experiment is because this NAE12 is very, very expensive. This figure that you are seeing here in the bottom, that alone costed us $35,000, just that one experiment. So we had to bite the bullet and do that experiment to produce those results to convince the reviewers, and finally we were able to do that one. So there are a number of functions of NAE, as I told you in here, and we wanted to further study how NAE causes defoliation of the cotton plants. The way it does is it induces this overexpression of fatty acid amide hydrolase genes in cotton, and this overexpression affects the hormonal balance. And because salicylic acid and jasminic acid production are completely rendered useless, then it makes the cotton plants more sensitive to ABA and also pathogen infection. And this is how the plants are able to defoliate, the strains are able to defoliate. And here in this figure, I'm just showing you the number of fatty acid amide hydrolase genes that are overexpressed in a defoliating strain as opposed to the non-defoliating strain. So that is to conclude. I just want to give you a story. In fact, in the University of California system, for all graduating PhD students, there is a competition in each of the campuses. So their entire PhD work has to be summarized within 3 minutes. And then if the winner will go on to get a presidential award, in fact, it's a very prestigious award. And I don't need actually 3 minutes. I have summarized all the studies in this one sentence that you see on the right-hand side. So I would like to thank all of my colleagues and also the funding agencies that supported this study. And before answering your questions, I want to leave you with this quote of a Zen master, Suzuki. And the challenge is to maintain that beginner's mind. Beginner's mind has a lot of possibilities. And if you're able to maintain that beginner's mind throughout your career, success cannot be too far behind. So with that, I'd like to thank you for this opportunity.
data that and the kind of conclusions that they have drawn. I would now like to keep this talk open for a few interesting questions if any one of you have, because let's not worry about lunch. Lunch can always be taken. It is good to have an interaction with a scientist like Professor Subbarao here and not to bother too much about lunch there. Professor Dubey first. Up mic लीजिए.
Exhaustive and complete story you have given. Two points in my mind. Whether this, what was the evolutionary process for the verticillium, whether the initially defoliating species came or non-defoliating species?
It is the non-defoliating strain that is prevalent because in nature, these are the only 3 hosts on which non-defoliating strains exist.
Okay, that means the virulence has been increased when it's become the defoliating.
Exactly.
So what was the time span required to differentiate these two?
Yeah.
Because you have not shown any physiological mechanisms, how the defoliation has been, the chemical factors which are required for the defoliation, but what happens structurally over there, what kind of the signaling is going, why they are detaching from what kind of the virulence pattern has been increasing, whether time span is reducing or increasing or not. That's
So we didn't put a molecular clock on this the phylogenetic tree because that was not the focus of the study. We didn't want to know. We knew that defoliation strains have been there for a long time. Even though they were only discovered in the 1960s, they must have been present much earlier than that one. So who knows when it happened, but what do they do once this virulence mechanism is acquired is what we discovered.
Okay, thank you, Professor Subbarao. Professor Chahal has a point.
Well, excellent work, and I must congratulate you. It's a very wonderful job you have done. This defoliation is really a problem in many crops, not in this particular host pathogen system. Verticillium is one, but other pathogens are also having the similar effect, particularly on field crops. The question coming to my mind was that we are facing in agriculture a lot of problem in cotton, a lot of problem in other crops. Say, for example, last year what happened that during this grain filling stage in wheat, there was a heat wave. Climatic conditions are variable and varying in different and causing a lot of damage to this crop production. Last year it happened in India that production was gone so down that it was less than even the previous year's total grain production. So it has very wide implications over my mind. It was coming when I thinking that not only this host pathogen system, but in others too. What is your take about this when we come to this climatic variations and how this can work there and can affect there?
Yeah, I mean, defoliation, as you know, it's a natural process. It's a natural process of aging. And it's only becomes a problem if there is premature defoliation. So that's what affects crop productivity. And you mentioned drought stress. Obviously, that is one of the factors that I indicated that also causes defoliation. So how do you resolve that one? There is no way to overturn a drought situation immediately. So we have to breed for drought tolerant crops, and that many groups throughout the world are doing it actually. And in fact, even when I was at ICRISAT, there was a large scale experimentation on drought tolerance in pearl millet as well as in sorghum. So that would be the way to go, I would think.
Yeah, Ashwini Parikh is at Punjab, sir, in Chandigarh. Ashwini Parikh is engaged in this drought tolerant development of drought tolerant varieties. Yes.
So Subbarao sir, it's very nice. You explain very nicely and through experimental procedures, but it is very interesting to know that why this virulent one defoliate, because if we logically think that the pathogens would like to survive longer time in the plant, infected plant.
Yeah.
So, but the virulent one is going to defoliate very prematurely. So there may be a other, you can say, a strategy plant, even pathogen is going to have it. Because I think the reproductive stage, if the plant is not there, so reproductive stage will be much more faster. So can you correlate these things? Because I am just is curiosity in my mind that how a virulent one is going to defoliate it much faster way compared to the, I
No,
think.
There is a very easy answer to that
Yeah,
one,
yeah,
I will tell you.
yeah.
So because only 3 crops defoliate, and this pathogen has to survive on those 3 hosts only. So when it defoliates, it is able to produce those microsclerotia on those defoliated leaves. So it is able to reproduce there faster, and that is its survival mechanism.
That's what. Thank you.
Any beginner's minds?
Yeah, exactly.
Myself, Dr. Bilal. So you said about evolutionary things. Have you, sir, done about selective sweeps? Have you seen selective sweeps have much role to play in evolutionary of this effector genes?
In,
Perhaps you-
in certain pathogens, exactly, selective sweeps do happen. But in this case, I don't think that is the process.
Since
It
you
was,
did comparative genomics, might
So-
you have given you some selective sweep ideas that this pathogen, rather than having horizontal gene transfer, might have a selective sweeps to evolve those type of PAMs?
no, I don't think so, because we found a perfect match with the sequences of the Fusarium oxysporum vasinfectum pathogen. So selective sweeps is a mechanism in several other pathogens, but not in Verticillium dahliae.
Any other questions? Yes, Robin.
So thank you,
You're welcome.
Dr. Subbarao. So your gracious presence and excellent talk improved the quality of this symposium and also annual meetup Indian Phytopathological Society.
Thank you so much.
Very simple question I want to know. After seeing very nice story, scientific story, what we have witnessed just now. So Verticillium, by virtue, it is a vascular pathogen.
Mhm.
And you have shown differentiated between defoliated, suppose isolate as well as non-defoliated isolate. Now, did you compare how fast defoliating isolate can multiply in the vascular bundle as compared to non-defoliating this isolate?
We didn't study that specifically, but I can only speculate. I think it ramifies, once it enters into the xylem, it ramifies much faster than the non-defoliating strain. And that's how it is able to cause that defoliation. Because the more it reproduces, the more NAE it is able to produce and infuse into cotton. And once it infuses NAE into the cotton system, that results in the overexpression of that fatty acid amide hydrolase genes. And that makes it sensitive to ABA action. Yeah.
Thank you.
Okay. Yes, Hamida.
So very excellent and clear information. My question is from the NEA. You said NEA was not sufficient to defoliate. So is it a quorum sensing molecule, because the pathogen is also necessary. So in Verticillium, can we call it as NEA is acting as the sensing to induce the pathogenicity or the defoliation?
No, it does, as I just told you, it results in the overexpression of the cotton genes. It makes it more sensitive. So that is the function of NAE.
So it's not with the lipid signaling, or because fatty acid hydrolase is also related here.
It is possible. It is certainly possible. But the direct conclusion that I can make is this one. So,
Thank you.
yeah.
Okay, I think there are no more questions, and I would like to thank both Professor Rakesh Pandey and Professor K.V. Subbarao for the wonderful talks on behalf of the Indian Phytopathological Society and on our own behalf. And I would like to request my co-chair, Professor Sridhar, to summarize and close the session.
So we had two very interesting lectures by the presidential address by Professor Pandey, and also Professor Subbarao has given a very exciting information about the genomics, especially with regard to the disease. And I have a small question. Is it possible to prevent the horizontal gene transfer?
No, I don't think so.
Any possibilities?
Nope. The horizontal gene transfer has been happening for millions of years.
Yeah, that is
From
an evolutionary process.
bacterial systems to pathogens or plants. And
Yeah, yeah.
in fact, I'll tell you, the race 1 effector that I told you in Verticillium dahliae, it was originally a plant protein. It came from the castor plant.
Okay.
So it ended up in Verticillium dahliae.
Yeah.
So how can you prevent it? No.
Okay. So thank you very much, sir. I thank both the speakers profusely for their excellent presentation, and also it has created a lot of interest to discuss with them, and I hopefully you can discuss the rest of the things during the lunch hour. Thank you.
Thank you very much. And in fact, when Robin sent the email to all the membership, I must have been the first one to reply to him that I'm attending this meeting. I was so proud. Thank you so much.
Thank you, sir. Dr. Krishna V. Subbarao, for your very informative talk on comparative genomics resolves historic puzzles in plant pathology. And now we'll honor Dr. Krishna V. Subbarao. And I request Dr. S.C. Dubey, ADG, Plant Protection and Biosafety, ICR, to please come on the stage to honor Krishna V. Subbarao, sir. And also I request chair and co-chair to join. And I request Dr. S. Chandranayak, organizing secretary, and the president of IPS and the secretary, IPS. Please, sir, please. And I request thank you, sir. Thank you very much. It's our pleasure having you in Mysore. And I request Dr. Rakesh Pandey, President IPS. Yes, sir. I request Dr. Chahal, former President IPS and former VC, MPUAT Udaipur. Sir, yeah, to honor Dr. Pandey. Sir, please. Okay. Thank you, sir. Thank you very much. Okay, sir. Yes, sir. Okay. Now I request Dr. Rakesh Pandey, President IPS, to present token of memento to our chair and co-chair. Professor Apparao Podle, University of Hyderabad, Hyderabad. Thank you, sir. And this co-chair as Professor K.R. Sridhar, Mangalore University, Mangalore. And I request our organizing secretary to present a token of memento to Dr. Harleen Kaur, Punjab Agriculture University, Ludhiana, Punjab. And to Dr. D. Parmesh, AICRIP Rice, Gangavati, Karnataka. Thank you, sir. Now we'll have a lunch break. I request all of you to, yeah, it will be a 20 to 30 minutes break. After that, I request all of you to be assemble here for the next session. Indian Institute of Sugarcane Research, Raebareli Road, Lucknow. And he is a scientist and ICR, Indian Institute of Sugarcane Research, and Hyderabad, and a senior scientist and a principal scientist, and head division of crop protection, ICR, SBI, and also he served as a principal investigator, AICRP on sugarcane, and a principal scientist, head and crop protection, Sugarcane Breeding Institute, Coimbatore, Tamil Nadu. And he has been awarded with many prestigious awards like MS Rugby Memorial Award in 2020 and Indian Phytopathological Society, New Delhi, and Sri Syed Ahmad Khan Memorial Award, Aligarh Muslim University Centenary Award in 2021, and the Sugarcane Technologies Association of India, Noel Dare Gold Medals in 2002 and 2022 consecutively, and Sugarcane Technologies Association of India Silver Medal in 2001, and so on to his credits. I welcome you, sir. I welcome you to the session. Dr. R. Vishwanathan. And this session is co-chaired by Dr. M. Krishna Reddy, ICAR, IIHR, Bengaluru, Karnataka. And is presently working as a principal scientist, head division of crop protection, ICR, Indian Institute of Horticulture Research, Bengaluru. And his area of expertise in the field of plant pathology, plant virology, molecular plant pathology, and biotechnology, host pathogen interaction, molecular diagnostics, and virus resistance identification and development of resistance genotype, epidemiology, and integrated virus disease management. And he has also been awarded with many prestigious award recognitions and so on. I welcome you to the session, sir. And today's session rapporteurs are Dr. Prakash TL, ICAR, IARI, Indore, Madhya Pradesh, and Dr. Amrita Das, Division of Plant Pathology, ICAR, IARI, New Delhi. Welcome you, ma'am. Welcome you, sir. We'll start with the keynote lecture, and I request our chair to introduce Dr. Apparao Podle, today's keynote speaker. Thank you, sir.
Good afternoon. My co-chair and my senior, Dr. M.K. Reddy. So, good afternoon, delegates. So probably we start the session with one of the eminent plant pathologist of the country, or globally we can say, Dr. Apparao Podle has contributed immensely on host pathogen interaction. So we are happy to, of course, this audience do not require so much introduction about Dr. Apparao Podle. So he is a man of such contribution, and served as Vice Chancellor of the prestigious Central University, that is Hyderabad University, and then assumed or had several positions to his credit, and also you must have you can see that he is one of the few scientists in the country who had been decorated with fellowships from all the major academies in the country. So that shows his contribution in the field of host pathogen interaction. And almost I know his work for the last 25 years plus. So he has been consistently working on that, and many of his students are in different laboratories elsewhere in the other countries. That also speaks about his or training the young generation on this new area of host pathogen interaction. And he, as I was referred that, he is apart from fellowships from academies, he has been adjunct professor in Charusat, Gujarat, adjunct chair professor in biotechnology, Asia University, Taichung, Taiwan, member of Indian Research Group, International Research and Training Group on Molecular and Cellular Glycosciences, supported by DFG in Germany. He is a visiting scientist in Department of Plant Biochemistry and Biotechnology, University of Münster, Germany. And he was also visiting scientist, Institute of Molecular Biology, Academia Sinica, Taipei, Taiwan. Postdoctoral fellow, National Science Council, Taiwan, at the Institute of Botany, Academia Sinica, Taipei, and many other places where he worked. And apart from that, the various, I am just reading out few honors. Apart from fellowship, he was member, Executive Council, National Academy of Agriculture Sciences, New Delhi, EC member, Consortium of Academic and Research Ethics Committee, University Grant Commission, adjunct chair professor, Department of Biotechnology, Asia University, participant Fulbright US India Education Foundation, sponsored by Fulbright Academic Administrator Seminar, November 2019, KC Mehta Memorial Biennial Award, National Academy of Agriculture Sciences, New Delhi, JC Bose Fellowship, Department of Science and Technology, President Association of Microbiologists of India, honoris causa, that is a DSc from Ram Manohar Lohia, Awadh University, Tata Innovation Fellowship from Department of Biotechnology, and he has more than 33 years teaching experience and completed more than 24 external funded projects. And even now he's continuing with many funded projects. I invite Dr. Apara Podile to deliver his the first keynote lecture.
Good afternoon, everybody. Yeah, you want to say something?
No, all the presenters, we request you kind cooperation. See, due to shortage and late starting of the session, we request all of you restrict to the time schedule. And keynote speakers will have 20 minutes to deliver talk, whereas invited talks 15 minutes, and the oral presentation 8 to 10 minutes. I request all of you to cooperate on this aspect so that we can successfully complete in time this session. Thank you. Now I request Pudli sir to go ahead.
Thank you, Chair Dr. Viswanathan and Co-Chair Dr. Krishna Reddy, distinguished professors and participants. I was thinking that I will do it in about 25 minutes. Now I'll try my best to do it in 20 minutes since Dr. Krishna Reddy indicated about 20 minutes time. There is something, the topic that I have chosen to discuss with you is to introduce some concepts which are relevant today, where we need to really think about how to understand the plant pathogen interactions to move forward. And that's one reason why it may take a little longer if it is only the second part of the lecture would be only about our work that we have done. So since I'm combining the both, I thought it may take about 25 minutes. Let me see if I can do it a little faster. So basically my interest is in today's topic is chitosands. Chitosands, we have been working for quite some time. And in that, we have done more significant work during 2005 to 2022, which has been the last 15, 17 years. And I will share some of those things with you. This is a slide which is quite familiar to most of the plant pathologists sitting here. The two types of immunity that we often see in the plants, that is the PTI and the effector-triggered immunity. And in between, you also see effector-triggered susceptibility. These are the two major immune responses that have been characterized so far, with so many details available for both PTI and also for ETI. The essential part of it is there are pattern recognition receptors and there are pathogen-associated molecular patterns. If these two things we are take home message from this slide is that. And this is one great review that has appeared, that appeared in 2019, the Tansley review, where the responses, because this pointer I will not be able to use, the responses that you find in the ETI and PTI, you can see they are very simplistic presentation of how the plant responds in PTI and ETI, and also with the viruses and all that is shown in this particular slide. And I will skip this slide. And this is a slide which I got impressed a couple of days ago when I was preparing for this particular talk, where there is a specific, there was a paper in Nature Plants that appeared in October 22, where a comparative genomics of 350 plant genomes has been done, and where the evolution of these pattern recognition receptors, particularly the extracellular ones and the intercellular ones, has been studied in detail. And a lot of information has been brought about by using this kind of a comparative genomics approach. Some glimpse of it we have seen from pathogen side, which was presented to us by Professor Subbarao in this morning. And this is the source for chitin. Primarily, the source that is used in the market for the chitin is the shrimp shells. And when you prepare the chitin, you get a pure form of chitin in this form. But this is an insoluble form, which is crystalline, and it is insoluble. And to make it soluble, most of the time, you will have to do a deacetylation process. When you deacetylate the chitin, you get chitosan. Of course, the degree of deacetylation varies, and then next things I will introduce. And when you deacetylate, there is a scope for solubilization of chitin, and it is the soluble forms of these molecules which can be recognized by biological systems. And as chitosans, as functional molecules, we are talking about the chitosans. When I go to the next slides, you will realize it is not simple chitosans. The degree of acetylation on the polymer or the oligomer, degree of polymerization of the oligomer, and pattern of acetylation of oligomer, all three of them are going to be crucial. And it is not easy in carbohydrate chemistry to synthesize selectively patterned molecules, and that is something which has posed a challenge for people. Not many people understand the importance of the pattern of acetylation, which I am going to highlight today. And after we understand the importance of it, how to develop that, but only biotechnological approaches can help us to do developing such type of polymers. And all of us know those who have contributed for sequencing of nucleic acid has got a Nobel Prize. So those who have contributed for sequencing of proteins also got a Nobel Prize. And in both the cases, it was Frederick Sanger who got a Nobel Prize for that. And this is something I was trying to tell you, that there is a degree of polymerization. You realize that is the number of residues that are there. And then the degree of acetylation, or you also express in terms of fraction of acetylation, the relative abundance of these N-acetyl glucosamine molecules on the polymer or oligomer is what we refer as degree of acetylation. And the pattern of acetylation is about the sequence of the N-acetylated residues and deacetylated residues, A residue and D residue, we call it AD residues. Now I take you to, because I said there are three things that I need to introduce. One is chitosan, one is immunity, one is the symbiosis. This is a classical symbiosis case where you find the rhizobium responding to a flavonoid molecule secreted by the plant, and in turn produces a lipopolysaccharide molecule the signal molecule from the nod factor that is coming from, and then there is a response from the host plant. And similar things like a nod factor, you find a mycorrhizal factor also produced by fungi. And the situation becomes complicated when it is fungi. When you have fungal system, when it is interacting with the plant can generate chitin oligomers, because the fungal cell wall is made up of chitin. And the mycorrhiza is secreting a mycorrhizal factor. And when these two are present, how the plant has to take a decision is a very interesting thing. Now we have to see what kind of informations are there about the lipid chitin oligosaccharides. They are primarily chitin oligosaccharides, which have substituted additional lipid chain attached to that. That is something which we need to understand. The entire recognition process between the plant and microorganisms is primarily focused on the chitin chitosan structures that are originating from the microorganism, be it a symbiotic organism or a pathogenic organism. And you can see the synthesis of lipid chitin oligosaccharides in rhizobium, which requires core genes involved in chitin synthesis, chitin deacetylation and acetylation, and other steps are required for the addition of the functional groups. So everything for a symbiotic organism, also these chitin synthases and chitin deacetylases are responsible on one side. And for the synthesis of chitin, also chitin synthases are responsible. This is one area which is still, I would say, a gray area where we have a lot of importance attached to the lipid molecules and mediated signaling in plants. There is some growing evidence that lipid-related molecules, which are most of them are shown there, and all of them are important in signaling between the plant and microbe interactions. There was a wonderful review in Trends in Plant Sciences in 2022 on that. And this kind of signaling processes are being studied throughout the world. Now, how the pattern recognition receptors at the cell surface and the nucleotide binding, the leucine-rich repeats in the intracellular, how these two are interacting with the molecules emanating from the microorganisms is being studied. One quick summary of it is there are two types of receptors that you find. One is the receptor kinases. The top panel is talking about the receptor kinases, which have a kinase in an intracellular kinase domain and an extracellular domain, which interacts with the molecules coming from the microorganism. Other type of receptors that you find are receptor-like proteins. They are called as RLPs. Varieties of these molecules are known to be present on the surface of the plants, which interact with a variety of molecules that come from microorganisms. And a small summary of it, I'm trying to narrow it down. What are the most crucial ones for plant microbe interaction? That is the receptors that contain the receptor-like kinases and receptor-like proteins that have an extracellular lysine modules. 3 tandem modules of lysine, when it is present, they are the ones which are important for recognition of the carbohydrate molecules that are coming from microorganisms. This is what I'm trying to show. You can see in all the 4 cases, in the first case, there is a nod factor with which it is when it is interacting with the lysine module containing receptor-like kinase, when it is interacting with a nod factor, it activates the symbiotic pathway, allows the colonization. And similarly, when it is interacting with a MIC factor, it allows colonization. And look at the extreme other side, the left side of it, you can see when it is the chitin molecule which is interacting, which is not the LCO, it is the chitin which is interacting with the lysine modules extracellularly, then you see that there is an MTI, that is what I was talking as a PTI. And the other one is also like a PTI when peptidoglycan, there's another carbohydrate molecule coming from a bacterial pathogen like Pseudomonas. So when pathogen is presenting a molecule, the plant is responding with immunity, and the symbiont is presenting a molecule, both are identical, and similar kind of a receptor is interacting with them. There is a hypothesis which has been put forward, which has solid evidence, solid ground for it. When the molecule coming from the microorganism has a lipid component, the lipid chitin oligosaccharide, then it activates the left-hand side, the common symbiotic pathway, CSP we call it. And on the right-hand side, we see the carbohydrate, only the chitin oligosaccharide, you find more of the pathogen-triggered immunity. That means there is something to do with the lipid attachment that is there to the signal molecule that is coming from the microorganism. So that's where I was trying to show the lipid-mediated signaling, which is gaining importance in plant microbe interactions also. So the molecule as attached to the carbohydrate moiety, and also the molecule without attached to the carbohydrate moiety, lipids are playing a very significant role. Lipid-related compounds are playing a significant role in plant microbe interactions. And this is a classical example which was shown in PNAS long time ago, some 10 years ago, which was shown where a chitin octamer, when it is interacting with the receptor, it induces dimerization of the receptor. And also it co-recruits an additional binding, chitin ester binding protein, CEBIP, is the chitin ester binding protein that has been shown by Shibuya's group from Japan. This is again, a simplistic picture of that. And now I enter to what we have been doing around this. We are working on a process, what we defined as transglycosylation. And it is when a chitinase enzyme acts on a substrate, it is supposed to cleave the molecule into smaller molecules. Here is a chitinase, which we have reported in Journal of Bacteriology a long time ago. And subsequently, we have done a lot of things around it, and also a couple of patents filed on that. And in this transglycosylation process, this enzyme not only cleaves the longer polymers into shorter oligomers, it can take the shorter oligomers and can synthesize a longer polymer. And synthesizing a longer polymer is always a difficult task in a chemical synthesis process. And we thought this is a wonderful candidate to do that. And that's where we concentrated on this particular enzyme. And we have done a lot of structural analysis of this protein. And initially we have done bioinformatic analysis. Subsequently, we have even obtained the crystal structure of the protein. And we have carried out several mutations where we could see there is an improvement in the transglycosylation process, whereby we can produce more of these transglycosylated products, more of these oligomers. Remember, I was talking about an octamer inducing dimerization. That is, longer oligomers are important in dimerizing or interacting with the receptor. So our interest was to produce longer oligomers. And this is the structure. And we recognize that there is an important loop structure, which is between the upper half and lower half of the protein, which is very crucial for the biological activity of this particular protein. And we also did some other studies where the substrate entry and substrate exit of the protein has been mapped. And our assumption was, if we close the exit site by introducing a bulky amino acid residue, if we can retain the enzyme substrate complex for a longer period, allow interaction, more of a transglycosylation products, and it indeed turned out to be the same. And when we did it, we could see that there is a significant improvement when we blocked the exit of the products from the protein for a longer duration. There was increase in transglycosylation. We also did some other alternative approaches where we have fused the different domains from different sources using some bioinformatic approach. We identified those domains and brought in, and we tried to further improve the transglycosylation process of that. Then what did we do with that? And we have been, by then we were already collaborating with a German group at University of Münster, Professor Bruno Moschbacher's group, and who were also interested in working on, in fact, they were the major group working on structure-function relationship of these chitosanes. So we also joined, because of our experience with the transglycosylation, we have joined that consortium of European consortium, which consisted of over 21 partners across the Europe and India. And a lot of work we have done on generating these oligomers from differently acetylated chitosanes, and analyzing those products and looking at those products and their capability to prime the plants and induce immunity in the plants. That is the approach that we have followed. And the two major contributions, first thing was from our own lab. That was in 2015, we have published. And after that, we have, as I said, in our collaborative effort with Bruno Moschbacher, that one was published in 2020. Where it involved more work, and we were collaborators with that, and we are partners of that particular publication of Journal of American Chemical Society as well. And this is how we analyze those oligomers that are generated after treatment of these chitosanes. We had to start with very well characterized, differently acetylated chitosanes, and we start with that, and then we start analyzing these products. And after treatment, and we do this size exclusion chromatography, we separate them and then try to test them. Each of these fractions 12 to 17 generated by this, and with the wild type strain, and with the chitosane, which has 61% deacetylation, they were analyzed through multi-top analysis. All that. Finally, we have narrowed down to DP8, degree of polymerization. Chitosanes with degree of polymerization 8, with 8 polymerization 9, DP10. And purified oligomeric fractions of showed a very significant increase in the activities of the enzymes related to immunity in the plant. And since we know O. Conrad is a very well-known researcher in plant priming. And we have looked at these molecules as agents for priming. And our assumption was, jointly, our assumption was that not only the degree of polymerization, I emphasized, and degree of acetylation, it is the pattern of acetylation on these oligomers also it could be important in inducing the biological activity. And in our case, it is the induction of the priming system. That is what we try to understand. And as you can see here, the monoacetylated residues on the of DP4 have shown excellent priming activity, whereas the monoacetylated residues on the extreme end and the completely deacetylated or completely acetylated residues do not have any biological activity. And taking clue from there, and we try to synthesize, making use of different kind of chitin deacetylases, we try to generate the oligomers of defined pattern. The deacetylated residue, you can see 4 different types of deacetylated residues that we have, a tetramer with defined pattern of acetylation we have generated, making use of these residues, and that we could see. Now you see the one which has at the non-reducing end, the acetylated residue has the highest priming activity, whereas the other ones which at the reducing end has very less of activity, which clearly demonstrates that the pattern of acetylation on the molecule is also very important in inducing biological activity. That's what we have shown in this particular paper. And this is a continuation of the same thing for the results. That part of it I have shown. And what we have also done is we obtained, Sharma is sitting in this audience, is my student. He has developed a process making use of those improved enzymes, a transglycosylation process, set up a transglycosylation process and separated out the oligomers prepared from that and made use of these oligomers and tried to test them in 2 different ways, as root dip treatment and or as a foliar spray. And foliar spray treatment induced elicitation response early and sustained for a longer period. This was also published a couple of years ago in carbohydrate research. I will skip these publications part. Yes, the conclusion of it, I would like to show that the chitin and chitosan fragments, the DP is important, DA is important, and PA is important. And if we can define and produce, identify, define, and produce the molecules, we are going to have the 3rd generation chitosans, which are going to, I will tell you what I mean by 3rd generation chitosans. I just wanted to, again, bring back the original concept that I introduced, a combination of this chito-oligosaccharide and lipo-chito-oligosaccharide. Recognition promotes arbuscular mycorrhizal association. When both of them are there, still there is a symbiotic pathway. How is that happening? LCOs, when present in a mixture of along with the chitin oligosaccharides, drive a symbiotic outcome. And this mixture of signals is essential for arbuscular mycorrhizal establishment, has been proven in one of the recent publications that has come in Nature Communications. That was not our work. I am trying to tell you that this is where the research is going on. And the cartoon form of that is a model of the chitin fragments and LCO recognition during symbiosis. That was these were the authors, Feng et al. In the Nature Communication. This is my collaborator's article. Biotechnologically produced chitosans with non-random acetylation patterns, that means defined patterns, differ from conventional chitosans in properties and activities. This is another thing that they have published. We are not co-authors in this particular work, but this is another work that they have published. This means there is a code. There is a code for the chitin. And to some of us, it may look like I'm talking about what I'm talking is a fiction, but it may be a reality tomorrow. A lot of things we have, there is a reason for us to believe that these things are happening. And there is a way forward for us to see how best we can produce these kind of molecules, for us to take advantage of these recognition process of the plants how the plants recognize these microorganisms and how to make use of that. This is what I was talking about. When people were not knowing much about these chitosans, the chitosan was applied. What they were using, about 40 kilograms per hectare, was used for crop protection. And second generation chitosans, where a little of deacetylation things were known and degree of polymerization was known, only about 4 grams per hectare was required. That's what the today's water-soluble chitosans that are available plenty in market are coming. And what we are talking about is the third generation chitosans, which are defined degree of polymerization, degree of acetylation, and defined pattern of acetylation. And what we require will be a few micrograms of that. A few micrograms of that when we have these products available in the market. And I would like to thank all my colleagues, my students who have been with me in this journey when we have started sometime in 2005, and we have ended up here as of now. And I would like to thank my collaborator, Bruno Morschbacher, and also I would like to thank the IPS Executive Committee and particularly Professor Chandan Naik for bringing me here to share our experience of working with chitosans. Thank you very much. I hope I stuck to the time.
You are on time, sir.
Okay, thank you. Yes.
Provide multiple treatments.
Multiple treatments are required. What we notice is it stays at up to about 3 weeks. Up to about 3 weeks.
I had dabbled with chitosan on Fusarium wilt of celery, actually. That was in 1996 or so. So it was not a very successful experiment. Yeah, that's why I asked. Thank you.
Sir, now, sir.
Yes.
Now there is so many places people are working on nanochitosan. And it's almost now it is a area of nanochitosans. Many people are trying to use this to induce signaling as an agent. And how far it will be successful in the coming days?
See, the source that they are using for nanochitosan is very important for us to really understand what it is going to do. In fact, we have worked with chitosan nanoparticles. We have made use of the chitosan as a carrier, and we have entrapped the herpin, a bacterial elicitor molecule, into the chitosan. And on that, we have a couple of patents already, and which we are also negotiating. In fact, we are, when I was sitting in the morning, in the audience this morning, final sanction letter from DST has come, where we are now working with an industry where this chitosan nanoparticle based approach to induce immunity in plant is being, has to be started. The final sanction letter has come along with industry. What I meant to say is, unless we know the source and the kind of chitosan with which we are working, it is not going to be easy to tell. We will get some effect. The effect what we get may not be the best effect that we are looking for.
Thank you, sir, for elaborating. And almost it is almost 6 months course work, course lecture. You have condensed into 20 minutes lecture. Thank you. And we have been hearing, and lots of, over the years, we see so many new innovations you are bringing in this field of science. Thank you, sir. Thank you.
Thank you.
I request chair and co-chair to present token of memento to Upper House, sir. Thank you, sir. Thank you very much for your wonderful interaction and presence. And I request next keynote speaker is Dr. Krishna Reddy M. I request Dr. R. Vishwanathan sir to introduce our keynote speaker now.
So next, as we know, next speaker is our co-chair, Dr. M. Krishna Reddy, and he is one of the pioneer in plant virology in the country. And he had his education from different states. And finally, he had his PhD from IRI under the guidance of Professor Anupam Verma-ji. And also almost he has worked extensively and throughout his career at IAHR in various capacities. And recently he is retired from the service, but he will continue there as an emeritus scientist. And to his credit, he has been recognized by various organizations and including postdoctoral fellowship. And his area of expertise is on especially on plant virology, molecular plant pathology and biotechnology, host-path interaction, and molecular diagnostics, virus resistance, and related areas. And he has been heading the important project, that a network project on plant virus diagnostics. And that almost he was heading the project where the project was located in different centers. And he has ably guided, and 2 EFCs he has guided. And also he has supported the plant virology work across the country. And now I invite Dr. Reddy to present his paper.
Thank you, Chairman. At the outset, I thank the organizers and the executive members of IPF. It is indeed a privilege to be in IPS and present a keynote address. And my topic is about the integrated management of virus diseases, especially in vegetable crops. And if you see, the vegetables are very important in the world. China ranks first, India is the second, and totally 73% of vegetable production takes place from China and India. And China and India contribute to the even vegetable seed production, which is the world meeting condition. The intensive cultivation and extensive cultivation of vegetables has several problems, out of which the biotic stresses, viruses are the major. And you are all aware that currently the viruses are taking significant losses in vegetable crops. And in addition to the climate change, which is influencing the insect vectors, which are the primary responsible for virus spread, and the trade in globalization of horticulture crops, especially vegetable seed and planting material, is also responsible for heavy emergence of vegetable viruses. Of the 15% of global production, if you see, plant diseases are the one responsible. Among them, the major one is the 1/3 is the plant virus. Though it affects the cultivation as well as yield and in terms of loss and the quality and marketability, often the farmers we recommend the pesticides to curtail the virus diseases only, to stop the insect vectors, but not the virus. Till today, we don't have any chemical which can directly affect the virus. As you see, the number of virus genus which are present in at least in the intensive vegetable cultivation in the Asian region, more than 32 genera are there, of which there are several viruses. All of them are not economically important. If you see the major groups of viruses which are divided, the ones which are transmitted by aphids, and the ones which are transmitted by whitefly, and the ones which are transmitted with thrips are the major ones. And to some extent, the nematode ones were very less number. But there is a group of viruses which are transmitted through seed and as well as insect vector, the central one. And if you see the number one group, whitefly transmitted, there are several bigoma viruses. The bigoma is a single stranded DNA, which is the whitefly mediated. It is in several vegetables is a serious concern. And again, another whitefly transmitter is the creamy viruses. This group of viruses are transmitted by 2 species of Bemisia, Trialodis and Bemisia. Both, one is glassos, another is the field whitefly. Both are capable of transmitting these viruses. And in thrips cases, the orthotaspoviruses are the ones which are major. Among the Asian countries, capsicum spotted are the ones which are a serious concern in several vegetable crops. And among the aphid transmitted viruses, cucumber mosaic virus, earlier known to be less important in solanaceous vegetables. Currently in Asia and also in some parts of Europe, the virus is becoming serious and its behavior is changed. Earlier, it used to express in the plant the symptoms what you see in the top, but currently it is expressing in the fruits, but not in the plant, and then leading to the total market yield loss. The another group of aphid transmitter are the partyviruses, which again very serious concern in solanaceous and cucurbit crops. And recently, the aphid-borne polariviruses, which are persistently transmitted, are again a serious concern. And often the symptoms are confused with the whitefly transmitted diseases. And there are few viruses which are mechanically transmitted or externally seed transmitted. Those are the three viruses which are earlier known: tobacco mosaic, tomato mosaic, and pepper mild mottle. But there are two newly evolved viruses for last 2 years. It's a very concern throughout the world, especially quarantine purpose, and there is a serious restriction on the movement of these viruses because of their seed-borne nature. The viruses, if you see in terms of effect of the climate changes we experience, the climate change not only affects the virus incidence, increase in virulence and the host range, and also the vectors, which are the major ones, will lead to the increased temperature increase, make the susceptibility to the insect vectors, in turn leading to new viruses, new strains, and spread in a newer location in extreme conditions. As a virus, if you understand the initial virus, the infected plant, from their transmission through insect vectors, lead to the healthy plant, either through pollen or through seed, and then again through insect, and the cycle continues. So here is the one where we need to emphasize. Many often people will say that in virus diseases, there is no control, it's only cut and burn or throw it. It is not like that. It is the nature of understanding the pathogen very important. And in this case, what we need to understand is the prevention, first step, and then the decision tools, and then solutions, how to implement them for the managing these viruses. As aware of as any plant pathologist, we know that host plant, the interaction, disease development is the host pathogen interaction, the three triangle. Whereas in case of viruses, it is the one which is the fourth factor, is the insect vector, and are the tripartite interaction, virus, vector, and host. And environment influences all the three factors. The fourth factor, which is very critical for successful development of a virus disease. And therefore, any attempts made for the managing, we need to understand and then intervene in each step how to prevent this disease development. As a first step, we to take in case of viruses, the first stage is the production aspects. In the nursery stage, seedlings should be grown under nylon net at the 40 to 60 mesh, because most of them are transmitted by the sucking pests, the aphid and several sucking pests, which are very minute. So the nylon net used, often people as a misconception, they use the mosquito net, which is a higher mesh, which is not useful, but the critical is the one which has 40 to 60 mesh. And then the early incidence of disease eradication will also reduce the subsequent spread in the crop cycle. And the agronomic like non-host barriers, especially cereal crops or tall growing crops, growing border, which can reduce at least the non-persistently transmitted aphid transmitted viruses. We have done extensive trials and adopted for many farmers that seedling production. Nowadays, the seedling produced under the nursery conditions, most of the nurseries are not scientific. They use the shade net for the multiplication, which is not a insect-proof net. Whereas any nursery practice should be under nylon net, which is 40 to 60 mesh, which are scientifically produced. And if you see the seedlings, the quality and in terms of the virus presence is nil in the beginning, so that the seedlings are protected in the early stage and will be having a healthy plants in the ground. Further, to develop the resistance or the strength in the seedlings, we need to apply bioagents, biopriming in the seedling stage, so that they go very effectively. And in terms of management, the manipulation of host and the arthropod vectors is a very important area. Now current research is change in the orientation and behavior of insect vectors to attract and repel the host plants. And there are several research carried, and we also have done some external work on this area, where the volatiles emitting from the plants are the responsible either attraction or repellence of the vectors, and that leads to the very efficiency. And there are ample evidence now, the different groups of viruses, if you see, the polariviruses, they affect the growth rate, alert production, dispersal, and fitness of the aphid vectors, which in turn, if you see this case in the case number 1, wherein the infected, the healthy plant, when infected, certain volatiles are generated, and these volatiles are recognized by insects, vectors, and they in turn feed on them. And then once they acquire the virus, again, a virus will modify this behavior of the aphids, will attract to the volatile produced by healthy plant, thereby it is going to feed on healthy plant, but not the on already infected plant. Once it acquires the virus, it doesn't go again to the infected plant. That is the mechanism how it is there. This is the how now current area of research, the identification of volatiles and change the behavior of vector. Already the using next generation sequence and then the meta-transcriptome analysis done with this 3 groups of insect vectors. There are several defense regulated genes which are upregulated and are downregulated when they feed. And these mechanisms, again, depending on the host phenology, host volatile, and genes which they are expressing under biotic stress conditions when the insect vectors feed or the virus enters into the host. The major things which are now happening in case of viruses is the one salicylic acid, jasmonic acid, and ethylene pathway. All these 3 are the responsible for activation or suppression or reducing the virus replication. One of the now earlier chemicals were used is the some BABA, the brassinosteroids, silicon, and BTH. BTH is the commercial formulation worldwide used except in India. But now Professor Podili has explained the chitosan to sir. Now currently chitosan is now has been used inducing against virus resistance in combination with other metal ions as well as with other stimulants of bioagents. So thereby it is leading to the expression, especially there are 2-way interaction which happens in the biostimulants. One is the repellence of the insect vectors, which are the primary spreader of the virus. And the second is in turn virus reduction is happening in terms of replication and the virus in the host plants. And these are the few examples showing that there are several reports which are already published in vegetable crops. And if you see the recent publication where bacillus has been shown to affect in the tobacco mosaic in tomato, wherein 3 genes of polybiosynthetic pathways and 3 genes of PR proteins are upregulated in treated ones as compared to the control. And there is significant defense regulation in these which are reducing the. In our case, we have done in chili wherein 3 viruses, the chili leaf curl, cucumber mosaic, and chili vinyl mottle virus, when we applied, salicylic acid and aminoliquefluence bacteria, and then a combination of microbial format, that is AMC. In all the cases, for up to 7 days, there is significant reduction in the virus replication or multiplication, but 15th day, there is a elevation of this. Though it comparatively it is control, it is 50% less. There is so this makes the determination of the critical interval time, which time to spray, so that they can be subsequently can prolong the host defense mechanisms. This is the mechanisms which I am highlighting. Then next we come to the resistance buildup. We have done resistance breeding for tomato leaf curl, wherein we have 4 hybrids developed based on this multiple disease resistance. It is not only resistant to tomato leaf curl virus, but also Phytophthora and early blight and bacterial wilt. And we have a chili again, 4 hybrids developed, which are resistant to cucumber mosaic and the chili vinyl, both the CMV and 40 virus. And we have recently developed 6 hybrids for chili leaf curl, which is a devastating for past 2 years throughout the country. And these hybrids we have now currently has undergone the field performance, and it has been demonstrated in 3 different states. And the farmers are very happy. In spite of the black thrip incidents, our hybrids are performed much better and able to have less incidence of chili leaf curl and higher yield returns in terms of yield and quality. And currently, though I am not dealing much on transgenic, the practically transgenic also been coat protein mediated has been developed, and it's a few cucurbit viruses, especially papaya ring spot virus and cucumber mosaic virus and Jubilee yellow mosaic virus have been done in the Europe and America, but in Indian conditions, no transgenic vegetables for against virus are available or not in practice. Commercially, till now, the virus-resistant chili transgenic lines are not available as on today. And current research is going on genome editing. We also initiated on chili and tomato for viruses based on CRISPR-Cas, wherein the elongation factor, which is responsible for binding the VP gene, and that we are modifying so that it can reduce the virus binding, which will ultimately the less virus incidents. Some of the examples already known for the genome editing for virus resistance are these are the tomato and in the cucumber and cassava and potato. These are already in the plants are ready, but till commercial cultivation, these varieties are not yet come. And when you come to the resistance part and then the overall management is the vectors, which are the very critical for these viruses. The control depends on the interaction of this, all the 3 types of behavior, non-persistent, persistent, and semi-persistent. Spraying is the most commonly used method, but it is not useful for non-persistent transmitted virus, because the moment to apply the insecticide, the virus is already inserted and the symptoms will develop. Then the next option is the preventing the landing and changing the behavior. That is the what we use, the reflective mulches or barrier crops or oils to spray so that they deter or the penetration and breeding behavior of the insect vector are changed. And some of these trials, what we have tried in case of chili, tomato, and capsicum, wherein the growing border crops with tall growing maize, then followed by silver mulching, then use of yellow and blue sticky traps, then application of selective pesticides in the initial up to flowering period only. No pesticide is recommended after flowering or fruit set, because the retention and the residual problem, and not only that, the incidence of viruses once the fruit set or the economic part is developed is less than 5%. The yield loss is not there, so there is no need of spraying these chemicals. And in addition, farmer do not keep quiet because he has a tendency to use insecticides continuously. What we advise them to change their psychology. Application of micronutrients and the seaweed extract or the microbial formulation, which in turn strengthen the biostimulation of the host, strengthening the immune system of the host, so that will take care of the production yield level, and there is incidence, even it occurs, it is less, not causing much yield loss. And if you see some of the trials, what we have done, if you see the border cropping, mulching, you can see the excellent yield obtained, 3 to 4 times higher than the normal ones. And it is same thing in case of chili and capsicum. The farmers are adopting in Karnataka and also Tamil Nadu and even in Maharashtra. And we also done in case of watermelon against watermelon viruses, and cucumber and muskmelon, and in bitter gourd. Now, bitter gourd is seriously affected by the whitefly transmitted viruses and the polariviruses, which are very serious concern. We have successfully shown, and if you see in our experiments for past 3 years, the application of aminoliquins and the seaweed extract and the neem seed oil has contributed higher in the production of yield, whereas the chemicals, acephate and fipronil insecticides which are used are not on par with this one. It is higher, these things. It is merely not that virus is not there, but virus incidence is reduced and the effect of virus is not seen much in these treatments as compared to the insecticides. Whereas the the aim for the farmer is the economic yield. If yield enhancement is there, even less incidence of virus doesn't make the loss. And the future of this is the emerging plant disease management depends on the progress developments in terms of disease surveillance and identifying proper cause and the spread of the disease, and then improved disease detection systems to use proper detection of viruses and communication and giving forewarning systems so that we can develop early information farmers how to take preventive measures and predict the plant disease modeling and decision support systems to develop in different agro ecosystems, how to use these management technologies, and the integration of all these technologies is required. No single technology or method can stop the virus disease. It is the integration of all methods from seed sowing to the perennial harvesting. That is therefore we can definitely curtail the viruses, and it is also too important. We need to integrate other systems of science so that we can bring effective management of viruses in future. And these are my collaborators and my colleagues who have worked with us. And thank you, one and all.
Thank you, sir, for completing your lecture in time. And in a nutshell, with all your experience, you are able to come out with a combo package for managing the viral diseases, especially in vegetable crops. So for either north or south where we face this problem, so I think your guidance is still required to manage these emerging viruses in vegetable crops. You can take 1 or 2 questions. Yeah, yes, sir. Hello, Tambi, brother.
Like such a wonderful lecture by virologist beyond the diagnostics and genomics. So practical utility, lot of attention you have given to the audience. This is congratulations to you. As I have seen personally, the good agriculture practices and insect control, vector control, if you can do, we can escape the viral disease. That is good. And you have mentioned very rightly. Elicitors, chemical elicitors, as already has pointed out earlier by you, this is not providing adequate protection for longer duration. We have seen, especially in fungal disease cases also, maximum 2 weeks. What in the case of whether we can recommend to the farmers use this, or this has to be integrated with biocontrolling agents or some things like that, especially for the virus resistance breeding. Personally, this is also my personal observations. I may be wrong, but most of the resistant varieties not durable for longer duration as compared to other fungal and bacterial diseases. So whether we are not focusing to the vector and virus virulence together, maybe our program breeding program. Why I'm asking, because maybe we have tuned the program at the national level, whether the tuning is required to focus the resistant breeding, considering the vector, not only the virus alone, or there's such a frequent mapping of the virus population based on the host differential. We don't have information, because in IRI working, I have the experience, 2 genome sequence, nucleotide sequence, new virus, Delhi virus, Assam virus, Andhra Pradesh virus. Nobody has testing them on the differentials, which can be used this information for the breeding program. We are liking in this direction. What is your opinion?
You have asked, sir, all questions in one, this thing. And my answer is entire topic. So the first question, what you mentioned is the disease management, the bioagents. If you have seen, I have mentioned the application timing of the stimulants and bioagents, soil, and also at 30 days and 45 days or 60 days, only 3 space, because initially we are recommending insecticides. I mentioned clearly up to flowering period, we need to protect insects. Later on, insects, even if it comes, the virus comes, the effect on yield is reduced drastically. But our psychology is the single plant is shown symptom, farmers will not keep quiet. They will spray every alternate day, which is leading problem for us in terms of resistance buildup in the insect vectors as well as poison, contamination, and environment in the accumulation in the food chain. So that is why our recommendation is insecticides to be sprayed before flowering. The economic part develops later, so residues are not built in the fruit formation. That is why biostimulants we are recommending after the stop of insecticides. They gives satisfaction for the farmers that he is spraying something. At the same time, these are not harmful and will build the innate immunity trigger, so that it is not full protection. It is some difference. Once the fruit formation is developed, there is nothing is required. What we say, economic part is developed, disease comes also, it doesn't make loss. That is the concept behind that. The third one, what you asked, resistance point of view. If you say resistant breeding as compared to fungal disease, virus is also stable resistance is there. But our way of defining resistance and our stable resistance is isolated or not. Often in a hurry or to jump into quick, we release a partial resistance and claim as a resistance, which breaks easily. That is one reason. The second one is, in case of viruses and cucumber, we have varieties which we have developed 10 years back. Still our chili varieties are stable, resistance is there. Whereas your question is appropriate for bigomoviruses, single stranded DNA viruses. The recombination development of virus is very fast. Resistance breaking pattern is very fast. So for this, even if you develop a variety for 3 years is excellent. I rate it as an excellent. But breeding is not a one-time stop point. It is a continuous process. As change in the strain evolves, we need to bring another line for that. So there is definitely we can increase the resistant pattern. Broad genetic base identification is the critical issue. As you pointed even the other day in the diagnostics, whether is it work in this. That is where critical areas are. We don't identify huge gene pool available in the wild species. Unless we bring more number of genes, then only we can get a stable resistance, which can we claim that yes, my variety can perform. That is the slow process which is lacking. We need to hasten up this, bringing more pre-breeding lines, bringing more genes from wild, so that we can get a stable resistance source for this. I think I answered.
Thank you, sir. We will take up the next lecture.
Thank you, sir, for your informative lecture. Next speaker is Dr. Vishwanathan R, ICR, Indian Institute of Sugarcane Research, Lucknow, Uttar Pradesh. Over to you, sir. So 1 minute, I will tell sir to introduce you.
And Dr. Vishwanathan
Yeah.
is my close friend and colleague, and we worked in several projects together. And he is a well-known scientist and especially contributed for sugarcane improvement and sugarcane viruses, and developed a lot of diagnostics. And his work has been recognized nationally and internationally, and he has been awarded with several awards. T.S. Venkatraman Award and Puttarudraya Memorial Endowment Award, Syed Ahmad Khan Memorial Award. And he has been also a founder and shown the evidence that in sugarcane, we all know sugarcane yields in India, whatever we are getting, it is the excellent yield. But he is the first time demonstrated all of them have virus. Without virus, there is a 20 to 40% enhancement of sugar yields when virus is not there. In this direction, he has worked the production of virus-free planting material and generating through tissue culture and supplying to sugar industry, and contributed vast in the sugar improvement program for past 5 to 10 years. I now invite Dr. Vishwanathan to share his knowledge for the audience.
Thank you. Thank you, sir, for that. And thank you. I thank Indian Phytopathological Society for inviting me to deliver this lecture, this August gathering. So I will be presenting on most on pathogen variation in a red rot, that is important pathogen. So of course I don't touch on sugarcane viruses for this lecture. So coming to sugarcane and red rot, so the both at least the host and also pathogen cannot be separated. So that is either in the farm, in the field, or in the research front. So that's the importance for we give to the disease. Since when the disease comes, it's almost causes 100% losses, up to 100% losses is possible. So that's what that we have been working on the disease for more than 100 years in the country. But still, of course, where we started, still we are at the same place. In the sense we have red rot epidemics, severe epidemics that we'll be seeing. And just one line on the crop scenario. And we know that now sugarcane India has been in a unique position. So that is India is occupying the #1 position in sugar production. And of course, the area wise, Brazil still tops in the world. And of course, they divert most of their sugarcane for biofuel, bioethanol production. And coming to Indian scenario, we also now India started exporting sugar. For the first time, India has occupied second place in sugar export and earning about 40,000 crores of foreign exchange. So that is a very big achievement in this sector. And apart from that, also India has at least the green energy program. And already we have achieved 10% blending of bioethanol in petrol. So though the target was fixed much later, but we were able to achieve the 10% blending, and we have to achieve or to reach 20% blending in another 2 years. Probably with all crop production and protection technologies, we will be able to achieve this target. Though it is a big target, so we will be able to do that. And in the present lecture, so my lecture will devote more on the red rot epidemics and also on the pathogenic variation and what was the work which we have done on omics, that is to genomics and the transcriptomic part of this pathogen, and also to for some aspects and functional validation of the pathogenicity genes. So as I said earlier, this both sugarcane and red rat are almost there together for so many years. The reason is that, and we have found that many occasions, we found the breakdown of resistance due to evolution of new pathotypes. So that's how we are continuously evolving new varieties. And also, but another important thing, that pathogen variation is also happening. But only way so far we are able to manage the disease also through varieties. So that's how we have released n number of varieties to manage the disease under field condition. And that's how that now even today, a variety can be released for commercial cultivation if it possesses high yield, high quality, that is high sucrose and red rot resistant. So still that we see maintain that very rigid strategy to manage the disease. And just if you see this overall, that last several decades, we had the red rot epidemics across the decades, but almost many elite varieties we have lost. And currently also this our variety, that is CO0238. Probably you must be remembering a few days before, Dr. Bakshiram was honored with Padma Shri for his work on evolving this particular variety 0238. This particular variety is even today is occupying more than 80% the subtropical region. That is, it is almost occupying more than 40% of the cane area in the country. So that is how we made a big revolution. But also we have found there is a severe red rot also in this field. And just taking you back and how this really pathogen variation, how sugarcane pathologists identify the variation in the field. And it is something like, evolution of light isolates. So they saw that when you people who have worked on Coltotrichum and related genera, maybe knowing that light isolates are and also dark isolates. In this case, we found, and mostly this when we see severe epidemics means there will be evolution of light isolates. So that's how we see that highly sporulating like isolates, which that's how we say that now we say that also hypervirulent isolates. When you have a collection of isolates, always you will see these isolates, some are on the top are highly sporulating, whereas on the bottom are almost a matte type of one. So they are also pathogenic, but they are less pathogenic. So this is how we find variation. So then these are all the petri dishes, we see these cultures. But to assess the pathogenicity, we wanted to have developed or identify a set of differentials. So that's how over the years we worked at the Sugarcane Breeding Institute, and we identify a set of differentials, and then we were able to designate the pathotypes then and there. Another important thing, and when since it is a vegetatively propagated crop, and we found this different varieties, and also even pathogenic variation also we found in the same variety. So we could see several isolates. That too, I could get more than 100, 150 isolates. But the testing pathogenicity in this crop is such a tedious one under field conditions, where we have tested at 117 isolates and then grouped them. Then finally we found still we are unable to group them. Both we conducted Karnal and Coimbatore, that is representing subtropical and tropical locations. Still, we could see that we could not categorize the isolates whether it's tropical or subtropical. But what we found, this variation was mostly attributed to the host variety and also place of collection, and to some extent on the host, the location of testing. So what we find in this particular picture, where many of the isolates I have collected, particularly from Tamil Nadu, and same variety exhibits so much of variation for the pathotype. And there we adopt a method called plug method of inoculation. So we introduce the pathogen at the bottom of the cane, and then we assess the linear development of the lesion. So that's how you can see such a variation we find. And also coming to another important aspects of identifying the new pathotype, how the pathotype emerges under field conditions. Really, if you see this particular case where we have identified a new pathogenic race, that is we normally designate them pathotype, that is CF12, that means Coltotrichum falcatum 12, and where that you can see that one, it is compared against the previously established one, that is CF06. That is the one which has been responsible for breakdown of resistance in the popular variety COC 671. So that was the ruling variety in 80s and 90s. So that's how the entire tropical region, this variety went out of cultivation due to red rot epidemics. So what we found that the new pathotype has almost comparatively higher virulence. Even among for several years, 7 years of testing also, you can see, overall you can see this new pathotype has almost in the last column, if you see, more number of susceptible reaction, indicating that the pathogen has slowly adapted and it is able to cause susceptible reaction on the many of the resistant hosts. So we tested with a set of varieties. So that's how we can find, even in a set of differentials also we could see, and this particularly the new pathotype is able to establish many, and we can see susceptible reactions. So this is how we found that new pathotype has emerged. And that's how even, of course, it also the other pathotypes also were collected, but they also behaved similar to the new pathotype. And this how that finally what the conclusion on this after identifying the pathotype was, so that when we remove the variety from cultivation, though this particular pathotype from that variety maintains virulence, but it is a moderate virulence, whereas the new variety when it succumbs to the pathogen, so where the pathogen evolves quickly and gets or even acquire more virulence. So that's we suggest it is found, we found there is a similar to post adaptation we found in sugarcane. And coming to these particular important aspects on the current scenario and the boom and bust cycle. So as we see in the past, several times we found here this was reported based on the examples of Bihar, where the several cycles of boom and bust cycle was found in sugar production that was correlated with the sugar red rot epidemics. So that's how then they have defined that it is closer to or analogous to vertifoliation effect. It was reported in potato. And a similar thing is currently we are facing the issue of boom and bust cycle. As I was mentioning with the 0238, this is almost has been adopted in more than 90% of the cane area in the subtropical region. So that has recently it has broke down. So that's how this was almost the newspaper reports. And that it's so much almost monoculture which we have followed that led to emergence of new pathotype. And of course, within the ASRP and sugarcane, almost we different centers we worked with this new pathotypes, where you can see this 0238 that is a host isolate for the host for the new isolates, and were compared with the previously prevailing pathotypes of the first three, that is standard pathotypes. And the last column, you see that almost the variety 0238 succumb to the almost uniformly succumb to the new pathotypes evolved from the variety. So that means the pathogen, almost a new pathotype has emerged completely to knock down the host resistant, whereas the same variety is still resistant to the prevailing pathotype. So almost similar reaction also you can see, but that is how that phenotypically also you can see. This is the reaction of the variety to the previously reported pathotypes, almost exhibiting resistant reaction. Whereas if we use that own isolates, that is adapted isolate in the host, it is totally knocking down. So this is how we found. And also this the grouping of the pathogenicity also we found. All the isolates from that is a top group 2, they are from the 0238. They are totally different as compared to the other pathotypes previously existing pathotype. So that means what are the pathogenicity of the new isolates are totally different from the existing pathotype. That's how the new pathotype evolved. So that led to the emergence of that highly virulent or hypervirulent pathotype. So that resulted in severe damages to cane cultivation. Of course, there are so many thousands of hectares have been damaged. So now that we are, of course, we are working on management of the disease also. And this how this led to the emergence of new pathotype. And here also we found the similar phenomena, that is host adaptation. The pathogen most the pathogen slowly are gradually adapted to the new host 0238 resistant host, and has caused breakdown, at least knockdown effect was witnessed in the field. So also monocropping also we found as another cause reason for faster adoption and also spread of the pathogen. So these are the areas which we have been working on that, and so far we have designated around 13 pathotype in both from tropical and subtropical regions using a set of differential host. And moving on to this from pathogen variation, we wanted to study this pathogen almost to genome and also transcriptome. That almost a decade back we started the work, and where we are able to establish the genome of 48.16 MB in size, and also with close to 12,000 genes we have identified. And also in the transcriptomes also we have completed. And also we found the comparative genome, we found this very close to maize and sorghum infecting Coltotrichum species. And you can see the transcripts also similarly you can find, and mostly they related to Coltotrichum graminicola. And other parameters we have almost this routine bioinformatic analysis we completed. And what we found these Coltotrichums that are related to the infecting sorghum and maize, the most grass infecting Coltotrichums we found, they are almost genomic as well as the transcriptomics. We found very close relation. And overall that our transcriptome analysis And we found that we are able to identify almost different sets of especially CSEPs, CASEs, and also the metabolites enzymes. And also then we after establishing genome and transcriptome, we went for studying the comparative transcriptomics. The first set we compared the virulent and avirulent C. falcatum, and the second aspect we went for again in vitro and in vivo, that implantar transcriptome analysis we have studied. Of course, this was the workflow. And this final results is that almost we have different resources in the omic approaches, both whole genome, transcriptome, and comparative transcriptomes of C. falcatum isolates, virulent and totally avirulent, and also implantar C. falcatum and sugarcane transcriptome. Of course, we are able to, this is data of the different omic results. And from the where we found around 12,000 predicted genes, around 12,270. And also in the host interaction, also you can see in the last, almost we could get more than 37,000. Among that, again, it was grouped the host genes and pathogen genes. So this work, some work is completed, some are still we are continuing. Add this, of course, these are the things of, again, we compared those host transcriptomes. Again, we found their close relation to sorghum bicolor and jia maize. And of course, we do reanalysis the relation that almost the picture may vary. And then further we studied in depth on the actually the plant cell wall degrading enzymes and also carbohydrate active enzymes. And that and again this related analysis we done for cutaneous spectate lyase, and that what we found, this stalk infecting coltotrichum, they have a specific pattern of the transcriptomes. That's what we found here. And they are found similar, all the stalk infecting coltotrichums as compared to the other coltotrichums. So what we found in the first, you can see that actually since sugarcane is having a hard rind, so what we found this again the pathogen, not the transcriptome, we could see or we could identify many lacases and peroxidases. That is almost they have been characterized to be the genes responsible to stalk infection. And also another important thing, the pathogen is typical hemibiotrope. So there we were interested to know what are the stage specific, that is biotrophic and necrotrophic specific genes which are the transcripts which we say. And there also we could able to identify their stage specific transcripts. And where in that also we were able to identify and several cellulolytic enzymes and cutaneous and pectin related enzymes, and that are essential for degradation of sugarcane cell wall, and they are considered to be important pathogenic factor in C. falcatum. Of course, these are the things we have studied later also with individual genes. And coming to this, actually the proteomics approach also, we started with comparing pathogenic and less virulent two isolates. And where this left one is pathogenic, where we found even the in vitro proteome also we could see more number of protein spots, and they have been critically analyzed. And you can see this actually that number of protein spots also very high in the virulent one. And then again the proteome was almost we subjected to MALDI-TOF, and we identified many proteins which are associated with the pathogenicity. And also, and then further, whatever we have done and so many other transcriptome analysis, then we specifically we looked for the specific that pathway genes, especially here on the melanin biosynthesis pathway genes. As we know that many of the hemibiotrophs, especially the coltotrichums, they need to have the aprosoria. Without aprosorial production, the pathogenicity cannot be affected. So there you can see that we used here tricyclazole, which is one of the known chemical which prevents melanin biosynthesis. And there we used the genomic approach, and many of the genes which are involved in melanin biosynthesis pathway also we found, as when we use the tricyclazole, and also when there is a very limited upper soil production, we could see there is a reduction in pathogenicity, and also the genes also, particularly this transcript level also reduced considerably. And we further confirmed with the real-time PCR also. And subsequently, we went on for this, actually the functional analysis using that is a knockdown approach using RNAi, and where the several candidate genes from our work were we pulled out, and then we studied where you can see a very clear the acyl transferase, this that is knockdown mutant. You can see there is a pathogenicity as progressively almost reduced as compared to wild type. So that means this is how we are able to demonstrate. Yes, there are many number of pathogenicity genes have been very clear, and we are able to identify very clearly. These are the genes which are responsible for the pathogenicity. And we can see the sporulation also. You can see the same when there is a knockdown mutants, the sporulation is growth rate is reduced, and also sporulation also reduced. And continuing the same aspect, another two genes also studied in detail, that is glucose transporter and sucrose non-fermenting fermenting one. Again, the similar response we found here also. The knockdown mutants fail to induce disease, or that later, of course, there is a disease development was there, but we were initially, you can see till 5th day there is a pathogenicity was less, and that's how this how we have identified the genes. Whatever we identified, not all, at least a selected genes where functionally we validated. And these are the coming to the conclusion in my talk. And there in the apart from the working on the pathogenic variation and field conditions, this omics approach also we found. First, initially we established a number of genes that may be playing a important role during sugarcane and C-Falcam interaction, and both the interactive genomics which were able to identify. And also the same our studies also we were able to identify many of the CSCPs, that candidate effector proteins, and also secondary metabolites, and we found their more of their presence during the biotrophic phase, and also during the later we found that membrane transporters and CASI is expressed during necrotrophy. And it is almost a similar report was we found in other coltotrichums. And coming to the last point, of course, we found C-Falcatum expresses 2/3 of the CASI during biotrophic and necrotrophic phases. So this is other things we found in these pathogenicity weapons in the form of secondary metabolites or in the form of CASIs or peptide enzymes, lysing enzymes we are able to identify. And what would be the final that future approach of this study, having confirmed the pathogenicity of many of these pathogenicity related genes? We are clear that now what we can we are planning to do that to identify the hotspots. When we say about when sugarcane is grown in almost more than 5 million hectares, and especially in Uttar Pradesh conditions where this cultivates, UP alone cultivates half of sugarcane. So there the sugarcane is grown everywhere, but this is also there. But we have to identify the real hotspot. And now we have almost a definite markers. We can say there's many of the genes we are able to identify, and now we further proceed on the comparative genomics, and by collecting the new isolates. Of course, we have already having 65 isolates from UP alone. So like that we will be proceeding, and also the further in the host pathogen interaction also further we have to proceed on the specifically on the susceptibility genes, which are in at least which we are already identified, and some of the gene susceptibility related genes we have to further validate, and then they may be used as a candidate for a genome editing also. Thank you for opportunity given. And of course, most of my work I have done at Sugarcane Budding Institute. Now, recently I shifted to Lucknow. Probably I will continue this work with a new team. Thank you.
Audience, any clarification? Yeah.
Thank you, Dr. Vishnath.
Thank
Again,
you
for a wonderful work. And we are the part of the networking projects. A lot of things we have revealed over there. One thing is still because your EDG, Dr. Singh is also discussing with me several times.
Yes, sir.
So maybe he's planning to have a brainstorming on the races and all these. Whether this is critically, we have to see whether the development pathogen part is changing or adoption, because you have already stated
Yes.
both the theory, which one is the dominant and really we can impress upon, because what differentials testing you are doing,
Yes.
this needs
Yeah.
thorough revision.
Yes, yes.
Because I have seen the column number 4 and last column is the same.
Yes,
That
yes.
means the same earlier already the variety having the same set of genes which are giving the susceptible response to most of the, we should have to test the new or original isolates and the new isolates with the same set of differential, then we have to see.
Yes.
This parts you have to strengthen. Nothing
Yes.
is a respond to this. This has
Yes,
to
yes.
be. Second one is the pathogenicity procedure. You have deputed someone. Now you are not free, but your guidance may be used. We want to see the pathogenicity in the plant, not in the this whatever we are doing. Because in case of rhizoctonia, I have seen what the inoculation in the bits, what we are doing, it is not matching with the field screening.
Yes.
Most of the times, the variety which are showing resistance or moderate resistant in the field, they are become susceptible where challenge inoculation and put them in petri dishes, the rice leaf or other stem.
Yes.
So same may be the case of the Coleotrichum also. So this has to be need a development of the pathogenesis procedures and protocol in the networking. Also, we are trying, but we couldn't do this. So you depute someone to do
Yeah.
this. That would be better. Thank you.
Okay. Thank you, sir. Actually, regarding the first question, the adaptation is the one which we found very frequently. And actually, when the variety is introduced to a cultivation, it takes few years. And when the variety, if the variety occupies the more area, then this almost the severity of the epidemics also more. This which we have found in every earlier that boom and bust cycles also. And earlier that when we talk about CO671, COJ64 or CO1140, any varieties. Yes. Yes, that is what. Yeah, variety. Almost, but what we coincidence, sir, when the variety spreads to larger area, even though there is a virulent isolates, but we got many virulent isolates, but they are not consistent in virulence. The reason is that the host is confined to a small area, maybe a small pocket, but when variety spreads in 1 million hectares or more than that, nearly 2 million hectares. So what is the pathogen has unlimited opportunity to proliferate. So that's how we found this when we say that epidemic buildup. So that is favored by the host and also the environment. And so that's what adaptation, the one which we found clearly. That's why we see even today, though we say this variety succumb to red rot, but still it is resistant to all the previous pathotypes. So that means the pathogen has moved away from the old, its own host, we don't know. But most probably it has come from the already existing host plant only. But that is some other host, it found this as a new host, since this is the one which is occupying the large area. And probably the environment, like last 2 years back, 2 years continuously, we had a severe waterlogging. That is another factor which is favoring disease buildup. And also the differentials, of course, sir, we also discussed during last May also. So that almost we are redefining the differentials so that we will reduce. And earlier idea was to include the clones from Saccharum, Officinarum, Barbary, like that. So we have, and also Spontaneum. So that now we will be removing the old varieties that of course then the ACRP we are taking care. But the inoculation, what mostly we do under field conditions. We take up the entire study under field. Now we know where we take up under controlled conditions, especially for pathogenicity. And of course, the canes, when we split open, we may use some covers, cloth covers and all as a background. Otherwise, we do in the field condition, and also we simulate the condition where it is favorable for disease development. So normally we take up during southwest monsoon in subtropical region. So inoculation will be done during August. Mostly we take up before August so that the pathogen has enough opportunity to cause almost pathogenicity. That's what we take up that appropriately across the centers, not only at Lucknow or Coimbatore. Other centers also we have uniform testing, and the culture is also maintained two centers, Coimbatore and Lucknow, we give the cultures to the respective ICRIP centers that we are taking care, sir. Thank you.
Dr. Vishwanath, one question. You identified some grass infecting ones. What is the reaction on this resistant types and this grass infecting cases?
But they are not that virulent, sir. Sometimes, mostly this pathogen is confined to sugarcane. And maybe here and there you may get a few, but otherwise they are not so much pathogenic, sir. Even in sugarcane also we get some virulent types, like which we get in the grasses.
Thank you.
So,
Thank
thank
you.
you.
Okay.
Thank you, sir. I request Dr. Krishna Reddy, sir, to present a token of moment also to Dr. Vishwanathan, sir. Thank you very much, sir. The next invited speaker is Dr. Mehraj Uldin Shah, Kashmir University of Agriculture Sciences and Technology of Kashmir, Srinagar, Jammu and Kashmir, India. I request our chair and co-chair to introduce our next speaker. Thank you.
Professor Shah, he is Associate Professor in the Department of Plant Pathology and Seri Kashmir University of Agricultural Sciences and Technology of Kashmir, Shalimar, Srinagar. So he had a rich experience in the field of pathology, and where he held various positions from senior research fellow to current position, and also his research areas are mycology and plant pathology, plant virology, molecular plant pathology, molecular diagnostics, host pathogen interaction, and biological control. He is continuing his activities, and also he has been awarded with several awards and honors in a short career, and also he is guiding different the students both in masters and PhD level. I now invite Dr. Shah to present his paper, and please try to complete in 12 minutes. It will help us to complete the session.
Thank you, sir. Respected dignitaries on the dais and off the dais, especially my teacher, Dr. S.S. Chahal, sir, other delegates, good evening. My talk will be to draft genome of Velsinomyces carpophyllus causing short roll of the stone fruits using two approaches, that's Illumina HiSec and PacBio NGS platforms. Before I will start actual talk, so I will give just brief introduction about the disease. It's a devastating disease in all the stone fruits, including peach, plum, apricot, cherry, and almond in the nut crops. And these are the major crops of the valley or in J&K after apple, which are grown on large area. So this disease have been, there are a number of people who had worked on this disease, but on the molecular aspects, it has been started from 2011 onwards. And at that time, when we started the work, there were just 5 ITS sequences in the database when we started to work. And right now, we have came across the different developments from characterization, ITS sequencing, and whole genome sequencing of the pathogen. And it has been published in 2023 itself. So this is just how we started from the collection of the samples, then isolation, purification, pathogenicity. And you can see from the below cultures that how much variable pathogen is in pathogenicity as well as cultural characterization. Then this was the basic work we started with when we tried to work on this pathogen. These in the upper diagram or figure, we can see 5 sequences which are marked as red there. They were in the database from different countries like Mexico, and other sequences are from our place, which are submitted in the NCBI. And based on the ideas, there was not much difference between the isolates of the pathogen. But then we switched over to see if there is some variability in the genomics or genetic level. We use some ISSR markers, and based on that, we could identify some differences between the isolates. That's the clearly shown in the dendrogram at bottom, and it's a cultural characterization where there is a visual observations on the differences in the morphological characters of the pathogen. Then this is the just a diagram of showing how we went from isolation, purification by single spore culture, then DNA extraction and sequencing by Illumina, HiSeq, and PacBio platforms in order to see and assemble the genome in hybrid on the basis of hybrid mode. That's PacBio and Illumina HiSeq sequences. They were mapped together to have a hybrid assembly of the genome, and then identification of the SSRs and their validation for identification or diagnosis of the disease in different crops. Then this is just how we did it. We took different isolates of the pathogen. Then based on the pathogenicity, we could identify that peach isolate that was highly virulent based on the symptomatology, based on the disease pathogenicity, and based on the number of days. In less number of days, it could cause diseases under the control conditions. So that's why out of 5 stone fruits, we have selected this peach isolate for the whole genome. Then DNA isolation, and then we have done the sequencing by outsourcing. This is just how we did it. We got the sequences in from this Illumina, HiSeq, and PacBio. Then there was a hybrid assembly, what I said. Then there was a pipeline quality check, then analysis of the sequences, and then submission of the genome to the NCBI under different accession numbers. This is just, we have deposited the genome in the available database of NCBI, which is available to others also. This is the just how we did it from the assembled this approach of 2 sequences when we did the assembly of the genome, then de novo assembly of the genome, then gene prediction model was performed using Augusta's gene annotation by 3-4 methods we did, so that we could identify which one will give us the best results. So this is the summary of the status of the genome. So we could identify from the genome total 130 scaffolds, and total genome size that including the gaps was around, it was 30 MB, and without gaps, it was 29.5 MB genome with the scaffold N50 that was having 662.3 KB this value. Then average scaffold length was around 117 MB, and then maximum length was 21.8 GB. GC content was 49.77%. Then in gene prediction model, we could identify total 10,901 predicted protein genes, these proteins coding genes, which were found to be by this Augusta software. And then total genome length, it was calculated as 13.7 MB, and the average gene length was found to be 1,263 base pair with maximum length of 31.8 KB. So this is the scaffold length distribution. We could identify that maximum number of scaffolds, they were having a length of more than 5,000, followed by 1,000 to less than 5,000. Then gene length distribution was also calculated, where we could identify the more than 5,000 gene length in maximum number of genes, around 5,000, more than 5,000 genes. And this is the genome annotation, where we could compare 3 softwares, that UniProt, COG, and PFAM database analysis, and followed by the Go analysis. Out of that, we could identify by one method number of genes, that was 6,331 genes by UniProt. COG analysis gave us 4,378 genes, and PFAM 4,272 genes. And then when we saw the number of genes which were responsible for different functions, we could identify the genes for the biological processes, molecular functions, and the cellular components. In this, we could identify maximum number of genes for the metabolic processes, cellular processes, in case of biological processes, and then followed by the molecular function, we could identify maximum number of genes that's more than 3,500, I think so, we guess. And then for cellular function also, we could get very good number of genes. And this is the just most of the genes which were found, that was 691 genes which we could identify. Out of them, they were having unknown, when we compare them with the databases, it was found that they were having unknown function because those genes are not known for the function. That was the maximum number of genes. And out of this, we could identify the genes for different functions using this software. Then PFAM analysis also helped us to identify the genes for their function, and maximum number of genes we could identify for the zinc fingers, followed by the protein kinases, chitin synthesis. I will not go one by one. So there was a huge number of gene counts which were identified in this genome for different functions. Then UniProt based annotation of the genes gave also a cytochrome P 450. For that, we could identify 697 genes. And then out of them, we found some genes which are with the function that they are responsible for the defense of the host system, because when the pathogen infect the host, so it develops the small spots on the leaf, and after 1 or 2 weeks, these leaves fall down, leaving a short hole appearance, which may have a defense system so that the infected portion fall down, so that there is no further spread of the disease. So those genes which we have found here in the other pathogens, they were also present in this genome as well. So this is these metabolic processes, genes responsible for the metabolic processes from the Welsonomyces carpophilus genome. And there we have also found a very good number of the genes which are present there for different functions. And this is the Venn diagram which shows the comparative analysis of various softwares by which we could identify various genes for their function. And out of total 10,901 gene, only 3,140 genes were common, which could be contributed by the these databases or softwares which were present in this genome. So this was the comparison of the genome after identification with the already available databases of the proteins. And out of 10,901 proteins, we could identify 9,427 proteins. We showed NR or non-redundant hits with the databases, and out of that we could identify most of the fungus with which it could show the highest number of hits is Pyronokita species, followed by Escochita and other 2 species. Pyronokita species shows 1,812 hits, followed by the other these species which are less than 1,400 hits. So this is the species distribution when we could identify the same, where we could see more than 1,800 hits with the Pyronokita species, which is already available in the database. And we could identify the most of the genes which are having the function, which are having known function in the Pyronokita species, were also present in this genome. And a total of, in addition to the other genes, we could identify tRNAs, rRNAs, and some pseudogenes in the genome also. So these are the tRNA distribution in this Welsonomyces carpophilus. Most of the tRNAs with their anticodon and these sequences hits, they were found on scaffold 1 and 2. So this is the diagram where we can identify.
Let's stop, please.
Yes.
Try to conclude.
Yes, sir. So this is where we could identify various predicted proteins which are present on the positive frame, negative frame, and with tRNA and RNAs, which are and GC content of the this Welsonomyces carpophilus. And in addition to that, we could identify some pathogen genes also in the this genome. So besides this, we could identify cell wall degrading enzymes, then chaperone proteins and all those. These are the proteins which are responsible for defense related mechanism in this host. And these are the SSR distribution. We could identify SSR markers for development of the molecular diagnosis of the pathogen. And out of that, we could identify 2,851 SSR motifs, which are spread across the genome in this. And this is the just little bit of work which we could perform for the diagnosis of that. So we could identify some SSR markers for identification of the pathogen from different hosts, except cherry. But later on, we synthesized more markers. And one of my students, she could identify some markers which can identify these gene directly isolated DNA from the host infected with the pathogen, and that she will be present in one of these other sessions. So this is just in addition to that, we could successfully identify this ATMT protocol. We could identify using the agrobacter mediated transformation, where we could identify some, we developed some mutants, and some mutants where they lost the total this pathogenicity, and some were having, in addition to losing these pathogenicity, we could identify some mutants which could sporulate but could not enter to the host with default, you can say, entry in the this pathogen. And so we can identify that there are some genes which are responsible for pathogenicity of the genes, but we can those mutants by mutation or insertional mutagenesis, we can default their pathogenicity so that we can use this for further management strategies. Thank you very much.
Thank you, Dr. Sir, for a good piece of work. So we can have a discussion maybe during tea time also. Thank you. Thank you for the wonderful presentation. Next,
Thank you, sir.
Next, we will invite Dr. Vinayak Hegde.
Yes, sir.
Yeah.
Please introduce him, Dr.
Yeah.
Vinayak Hegde, ICR, Central Plantation Crop Research Institute,
Yeah.
Kasaragod, Kerala.
So Dr. Vinayak Hegde has close to 30 years of experience in ICAR. He worked in different institutes, starting his career in CPRI Shimla. Then he moved to CTCRI Trivandrum, and then now last 10 years plus he is working in CPCRI. So he has worked as a principal scientist, later as a head division of crop protection. Of course, he is continuing that position even today. He had several awards and medals to his credit, and that also includes IRA Merit Medal for outstanding research work during for his PhD during 1994, and also US Gold Medal for highest OGPA in MSc Agriculture. And also the academic and the research contributions are immense, especially in the tuber crops and also in the plantation crops. And now I invite Dr. Hegde to present his invited talk, please.
Respected Chairman, Dr. Viswanathan, and Dr. Krishna Reddy, and esteemed delegates. And I'm quickly present about the managing recent outbreaks of foliar fungal disease in arecanut and coconut. I have taken this topic because there are a lot of news recently about the arecanut as well as coconut in some parts, mainly the arecanut leaf spot. I think there is no need for more introduction. These are the two crops. I think every one of us knows how they are important for each, especially in India, all the socio-economic regions. They are our cultural activities also. And they are the major, especially in arecanut, more than 50% of the world production we are leading, as well as in coconut, around 30% we are producing. These are the coming to arecanut scenario. Now arecanut is increasing area, being a cash crop, and returns. It is increased over the years, increase in area, and these are the major places where the arecanut is now being grown. These are the diseases which I would like to take arecanut, as there was not much disease problem except Phytophthora. As we all know, Phytophthora fruit rot was the one of the major in the arecanut problem was there. then Ganoderma, and yellow leaf disease. These are only 3 diseases were the major, and especially Phytophthora disease. That was the only one where the farmers were had to go for spraying during rainy season. That was the major issue. However, in recently we have seen that one of the minor disease which was there, which was causing leaf spots, now that became an outbreak type and caused a severe disease. This is the arecanut leaf spot disease. This became a very severe, and in many of the, this started not only leaf, or even on the nuts, it started, infection started, and severe dropping, and farmers lost around 50% within 2-3 years. Many farmers have lost, even up to some farmers, even up to 80 to 90% loss in the yield. It was recorded. So that's why then we started studying about this disease recently only. Though there was some report, earlier studies were there that leaf spot was caused by mainly this Phyllostricta species and Colatotrichum, Helminthosporium, Curvularia. But these were all, though reports were there, some studies were there, all are minor disease, and even farmers were never used to bother. Only in the nursery as well as in the some juvenile farms, they used to be there. And there was suggestion also that time, studies conducted at CPCRI as well as in the universities. So it was suggested that time Mancozeb used to be in 80s that time. Afterwards, there was no much problem, and farmers never used to bother for this disease, leaf spot disease. However, recently, which appeared, especially in 2016-17, in the Jumpai hills of Tripura, there was a sudden outbreak of disease, which we came to know. So we had to visit it and then checked. There was complete drying of leaves and this one. Then when we checked, it was due to leaf spot disease. When we analyzed this and checked Colatotrichum gliosporitis only, then we try to understand how come it is there that much leaf infection, because in the traditional area, we were getting only the mainly Phyllostricta, and it was not a very serious. And Colatotrichum infection was there only in the inflorescence, where we used to get the dieback disease that time. So we started studying, and we found that time, even the species level, we tried to identify where the Tripura. So it was the epidemic, it was Colatotrichum siamensis we got based on the analysis. Later, when 2018 onwards, I suppose during 2018-19, there was a severe incidence in the traditional, this arecanut belt, especially Tirthalli and Chikmagalur area. Severe infestation of this leaf fly started. This is the, we can see earlier, this is the common leaf spot. Even the farmers used to know in the left side earlier, we used to see like this. This is not a major issue at all. Lower leaves, few spots used to be there, and farmers also never used to bother. But whatever we are seeing now is starting with like this with yellow halo, and whole leaves becomes later suddenly yellowish, and it will become dry up. Leaves become sentient. This is the severely affected gardens. You can see how severe it will be. It was first noticed in Chikmagalur area, where we have seen there in the coffee area where they were planted arecanut in between. So that severe incidence was there. Then these are the now when we check, these are the areas where it is present in India. This Colatotrichum leaf spot, I can say it is not the Phyllostricta mainly. Phyllostricta used to be there everywhere, leaf spot. There was not a major issue at all. This type of yellow halo and this type of spots now we are finding in these areas, arecanut growing areas. So as per in the Karnataka, immediately there is a lot of national issue it became, and state level, all this one, then there was a estimate that around 42,000 hectares within short time, it was found. Of course, now again it's increased, slowly spread. So it is around 50,000 hectares maybe there now affected with this one leaf spot disease. So this is the places we visited immediately and try to find out what could be the reason for this and isolating the pathogen. So this is where we can, the severity of the diseases, we have noticed sudden drying. So this is during end of the season, we can see from the leaf, we got mainly whatever the spots we got, all we try to isolate and check the samples. Most of the places we got this Colatotrichum, and all these cultures, many isolates we try to, and culturally and isolated colostotrachum it was giving. Later we try to understand why sudden this one with the different genes as reported now as colostotrachum gliosporitis is a complex they're telling. So we followed the same procedure to identify the species level with the different multilocus sequencing. So even the pathogenicity, we tried to establish whether it's really pathogenic. So when we isolated and pathogen, the re-isolation also, we could able to get the same species. So this is the inoculated leaves and re-isolation also we could confirm. Then when we did the analysis, we can see here, this is mainly emerging type, all were representing this colostotrachum, as well as this is all in a separate group. This earlier, which we have getting colostotrachum type spots also was there, but these are very few. But most of this one, whatever we have isolated, this all belong to the complex. Mainly we got colostotrachum kahave. It was representing to the colostotrachum kahave group. So this is then when we searching this in the recently also 2020 when we are working this only, we came to know the publication was there from China in plant disease that there is a news this colostotrachum kahave one reconnect. There is a first report was there. Then we checked the same type when we are also analyzed. Our results also showed that it is colostotrachum kahave sigavar. That's our analysis matched to that one. So this probably this could be the one of the reason. Of course, there are a lot of work we still have to do. How exactly weather conditions we are working on that. What are the predisposing factors, nutritional, because we have started with that and we checked that most of this garden potassium is very low because most of this where the disease severe, there was they were mainly using organic. This one, there was no nutritional problem was also there. So it has flared up the disease. So immediately we have suggested as the ad hoc recommendation to go for phytosanitation and fungicides. We initially first when I 2016 as well as in 2018 initially when I went, we told that go for this carbendazim plus mancozeb because that was the one that time it was reporting for the colostotrachum disease. When earlier when I was working in YAM, we used to get colostotrachum anthracnose. That was the best fungicide. So we recommended to farmers to go for that. Then they told after a few days, sir, it is not working. We have to go. So then we started with hexaconazole and others also. So later we started some farmers to go for in alternative way, different triazole fungicides also, as well as this one immediately now we have suggested to go ahead with this, though it is very difficult to go for phytosanitation as well as community approach, because even arecanut to go for phytosanitation, it is a major problem as we know, go for removing the lower leaves, dried leaves and removing, and it is very difficult to eradicate the pathogens, the established, especially in that type. So we told for that's why the state government has also come forward to sponsor for the DOTs as well as for the chemicals. Now some compensation also they are asking for the farmers who have already lost more than 50% of their yield. So community approach also has been suggested, whether we have to apply drone. That is still, though few farmers and companies have started drone spraying, but we have just initiated whether there is a deposition proper will be there. That studies are not there still. And even though many of these, this also ad hoc we have recommended, as far as the arecanut is concerned, there is no label claim as of now still for these fungicide, triazole fungicides, except odomixture. Most of these fungicides are not having label claim in arecanut. And nutrition, these are the integrated approach we have suggested. And in coconut also, compared to also, these are the major areas in coconut in India. So these are the only, earlier we used to have mainly bud rot, Phytophthora, root wilt and Ganoderma. Wherever we see, these are the major pathogens we used to study. Now, recently, we are seeing that there are different types of foliar diseases were coming. So like this has become recently in northern Kerala. So we try to complete. This type of symptoms we have noticed, and we try to isolate. And we found that this is mainly the Lasiodiplodia infection was spreading in most of the this one, though earlier it was very certain areas in Pollachi, it used to be a problem. Later it started starting attacking the coconut in different areas, even the nuts as well as in the prawns, and we started finding the different species, this Lacidiplodia also, Lacidiplodia theobrome and Neodictonia penacum we saw in Kerala, northern Kerala part, where there was an outbreak of this type of disease. Whereas in case of Tamil Nadu area, which we have studied that also, and spindle rot, some of the area where we got that suddenly the spindle rot symptom was also noticed, which was not a bud rot of Phytopathological, but it was used to be used by the this person. So this recommendation we have suggested for this also. So as of now, there is no much problem. This disease now in the endemic area, especially that crown rot type of symptoms, though leaf blight is still a major problem in many of the places as well as not. Even in Pollachi and Coimbatore area, it is severe, as well as in other Karnataka places also. And we have seen that even many places, the pond area is also affected by this same fungus. So that is also causing a major problem. And even the spindle rot, especially in Tamil Nadu, many area where dry place is there, where Phytopathological may not establish that much. In that area, spindle rot, when we checked, it was found to be due to Lacidiplodia. So these are some of the two pathogens what I feel are emerging in the last few years as a major problem. This is also in some of the areas, 7 years old garden. This in the below 4 years old in the Tamil Nadu. And this is also in some of the area where especially DJ hybrids, which have become popular in certain area in Tamil Nadu, they are planted, where they saw that 50% of the incidence of this, and well as in COD around 20% incidence of these varieties, in these two varieties. So in the dry spindle rot was become problem, we just checked, and all this we used to get mainly this Lacidiplodia, and we tried to analyze also whether they can have a group. We thought Lacidiplodia theobrome. There are two types of, we grouped the culture based on cultural morphology as well as spore. So, and even the other isolates we could get through the genomic, this one as parva and pseudotheobrome isolates, also some of the variants we have got. So coming to the molecular identification of the pathogen also, we have identified and found that there are different variations, and pathogenicity also happened. This is the only conclusion, is that these two pathogens are, though very minor earlier, and we never used to focus much, now has emerged as a major problem. And still now we have to work out what may be the reason. And as you told, whether varietal, this one problem is there, or any other host or nutritional problem. So this has made this outbreak of these diseases. We have to find out. These works are going on to go for the develop whether we can have a forewarning or other decision supporting system. That is the demand by the department also. Thank you. Thank you, sir.
Thank you, sir. Thank you, Dr. Hegde. And of course, this important work you have covered, and it has taken more time, but still lots of work you have covered. And really this, whatever the work done at your center is most rewarding, and to solve the farmers' problem. Thank you. We will have more discussion during the forthcoming dinner time. Thank you. Thank you.
See, especially this leaf spot, what is happening, entire Sirsi and other belt.
No, Sirsi area also, certain area it is started spreading. Slowly it is spreading. Initially
it is spreading entire now Karnataka, wherever Aracanet is grown. And the farmers are more concerned. Earlier we had a Phytopathological problem, but nobody is talking on Phytopathological now. Everybody is worried for this.
Yeah, that is.
So you need to now popularize this technology for spreading management side, both coupled with nutrition, because farmers are not aware that this is a problem coming, coupled with nutrition.
Yeah, that's what we have given complete integrated approach. They have to follow. Simply spraying fungicides will not help to control the disease. So even many places already we have taken a lot of campaign now. Even the village
simultaneously, drone you can advise, and also you can also initiate a pilot program for demo trials using a drone, because reaching the Aracanet farms is very difficult task.
Yes, we have thought of that also, drone also, as well as even the DOTI based spraying, that carbon fiber pole. Many farmers have started using, and the department has started giving subsidized this one to farmers, that carbon fiber pole. Many farmers have started using that for spraying and harvesting. Okay, thank you. Thank you. I thank all the organizers for providing me the opportunity. Thank you.
Sir, I request chair and co-chair to present a token of memento to Vinayak Hegde, sir. And I request Dr. Mehraj Uldin Shah to receive memento. Thank you very much, sir. Thank you, Dr. Mehraj Uldin Shah. Next, I request oral presentation participants to get ready. All of them. The first presenter is Dr. Rakesh Kumar Singh, RVSKVB, College of Agriculture, Indore, Madhya Pradesh. Yeah. I request all the participants to download their presentation in the system.
Well, you can see I'm not Dr. Rakesh Kumar Singh, but I'm Dr. Bhabesh Dada. And one actually invited speaker was not there, so they swapped my talk into this one. So I'm Dr. Bhabesh Dada. I'm from University of Georgia, an associate professor and a vegetable pathologist. I'm actually
trying to complete in 8 minutes, no?
Yeah, sure. I'm a bacteriologist. I'm gonna tell you a story on basic and applied research on onion beetle diseases. All right. Okay, to start with, onion production. Good news, India is now the largest producer of onion in the world. We just surpassed China in 2021. So if you look at, I do not know if you don't, the pointer doesn't work here, but if you see, we do have a lot of arable lands and we can grow a lot of onions, but the productivity wise, we are not number one. Productivity wise, we are number 3. So what affects the productivity? Productivity is affected by marketable yield. And most of the losses in onions are because of bacterial diseases that can occur at pre-harvest or at post-harvest. And among the top onion producing states, you can see Maharashtra, followed by Madhya Pradesh and Karnataka and so forth. So onions, onions are so important, they can also make a regime change. So this is happened in 1998, high Indian prices led to government defeat into 1998. This is Times of India newspaper cut. So what are the common bacterial diseases in the world? You can see there are nearly 21 common bacterial diseases in the world. Out of those, 4 of those which I see in the arrows, which my lab has named and characterized. They are Pentoa agglomerans, Pseudomonas verdiflora, Pseudomonas allivorans, Pseudomonas cornifications, Phytopathological secola, and Ruxia lepidensis. But out of those 21, which are most important around the world, including India, is Pentoa ananaris, causes center rot, and Burkholderia cepacia that causes sour skin. Both of these pathogens or pathogen complex can cause at least 5 to 10% yield loss. If you account for the millions of dollars of losses to it, it comes around $125 million in losses annually. All right. Some of the important symptoms of the diseases. Here is the center rot. This is how it looks, caused by Pentoa species. This is bacterial streak and bulb rot caused by verdiflora and allivorans. This is what we discovered, and we have named this pathogen. This is sour skin. You might have seen this in your kitchen. When you cut open the onions, the onions may appear very well outside, but when you cut open, you'll see these rotten spots. This is yellow bud disease, very new disease, looks like a banana leaf, but it is caused by a Pseudomonas cornifications. We named it. This is Xanthomonas alii, very important pathogen down here in India, especially in warmer climates. You will see this pathogen. Onion bulb decay. How many of you have seen this while cutting your onions? This is another important pathogen. Burkholderia gliderella causing slippery skin. So more or less, you see all sort of onion rots. Now, question is, do all bacterial rots follow same pathway of infection? Of course not. Most of my lab, most of the work which I have been doing last 10 years is on pentoananatis and pentoagglomerans complex. They cause sun rot. It is one of the most important bacterial diseases in the world right now. And these are to my students and the postdocs who've been working on this pathogen. The particular beauty of this pathogen is that it has wide host range. It can affect, and it can survive on different plant parts. It can survive on different insect vectors, and it can survive on weeds. It has a multiple life cycle. So in general, we know bacteria infects plants in different ways. And one of the most important ways is using secretion systems. So here are the examples of type 1, type 2, type 3, type 4, type 5, and type 6 secretion systems in bacteria. Do pentoia utilize these? Not at all. They don't. They do not follow normal bacterial norm. So what they use? They use phosphonates toxins. How many of you heard about phosphonates? They are called herbicides. You know, we do have phosphonate group of herbicides. These bacteria utilize herbicide to kill plants. So this is a work done in my colleagues' lab, where they have identified set of 11 genes that produce these toxins and kill the plants. My another collaborator have actually isolated the phosphonate toxin and inoculated into the onion bulb. Look at that. What happened to the onion bulb? It decimated, it rotted. Now, this is my student, what he did. Well, you know, when you cut onions, what appears? You'll see sulfur compounds. The tears comes out, okay? The bacteria, when it infects onion, it has to survive the sulfur-rich onion environment. Okay, so what we have found that the bacteria needs to contain certain thiosulfonate tolerance gene cluster. Okay, it has to survive in onion in thiosulfonate rich environment. If the bacteria contains that particular tolerant cluster, it can survive in onion. So this is a nice study where we looked at we named this particular cluster as ALT cluster, allicin tolerance cluster. And we're utilizing lot of mutational analysis and functional analysis. We proved that this cluster is very, very important. Okay, question is hybrid cluster, which I showed the phosphonate cluster, and ALT cluster is present in all onion pathogenic bacteria. Let's see. So this work done by my another postdoc, Dr. Zhao, what she found there is another pentoia that utilize another sort of phosphonate. So there's one phosphonate called hybrid, which is utilized by Ananatis, but this particular phosphonate is produced by Pentoa stewarti endogenous, and we named it as Hellophos cluster. And you can see this cluster, when we knocked out that particular gene, you can see the Hellophos, it lost its function. It did not cause any disease or any lesions on the red onion scale. Okay. Now this cluster is also present in Pentoa alii, another bacteria. All right, so this is what it is. It is present in Pentoa alii. Now, here is our model. What our model is saying, for a onion infecting bacteria, for a successful onion infecting bacteria, it has to have two components. First, it has to have a phosphonate toxin. And second, it has to have a thiosulfonate tolerant cluster. So phosphonate toxin kills onion. When you kill onion cells, sulfonates comes out, and in that environment, the bacteria has to survive that high toxic environment. To do that, it has to have a thiosulfonate cluster. So this is successful bulb infection. This is pentoia, and this is onion cell. So when you have hybrid and Hellophos phosphonate clusters along with ALT and allicin tolerance, there will be successful bulb infection. Bulb will rot. Well, in case of there is another situation where pentoia can infect the bulb, but it cannot rot it. It can cause a little bit of lesion because it lacks ALT cluster, because it has to survive thiosulfonate rich environment. Okay, no infection. When infection won't occur, if the pentoia lacks those two important virulence factors, thiosulfonate cluster as well as allicin cluster. This is our model. Now, is pentoia is a hybrid or a Hellophos clusters is universal to allium cepa or onion? No, it is not. Which we have shown, my another PhD student have shown that For leek and spring onions, this is leek and spring onion, hybrid and helper cluster, both are very, very important. I don't have a pointer here, but I can show you here that when we used single knockouts and double knockouts of those phosphonate clusters, we actually removed the function of this bacteria to cause disease on leek and bunching onions. So these phosphonate clusters are also important to cause disease other than onion. Okay, now my research topic is basic and applied aspects. What should we do? Now, when a grower comes to you and asks you what I should do, you cannot tell them that, hey, we are breeding for resistance, wait for 10 years. You have to tell them, give him something. So I also do a nice chemical assessment. In chemical assessment, in what we have found, most of the copper products, if you apply them at bulb initiation stage, when the bulb swells, or that particular stage, then that particular stage is critical in reducing bacterial internal rot. You can see all the copper products work, although there is some level of variations, but most of them work. Regarding software products, LifeGuard, I don't know how many industry folks are here. LifeGuard is a software product that works. Now, one more thing which I have started in my introduction, I mentioned that Pentoa annularis is a thrips vectored pathogen also. In this case, we have shown when we don't have any thrips, look at the leftmost graph, no thrips, then your copper products will work. When you look at the right graph, when you have thrips, copper product will not work. What does it mean? The thrips, despite whatever you spray, thrips can get into the onion bulb and feed and transmit the bacterium. Okay, this is a nice field study, very extensive field study, which we have shown in the field. When you have a thrips program plus copper program, you'll have a better success compared to only copper program. How much time do I have left? Oh, you're just testing, sorry. This is another trial where we looked at thrips management program overlapped or overlaid with the copper program, provided our best management over here. Okay, another thing, do we have to rely on copper only? No, not at all. We can make small cultural changes that can actually give you the dividend. This is, look at that onion. That is called a matured onion when you have a dried neck. Okay. When you have a solid neck, that is not matured. For a successful bulb infection by a bacteria, you do not need a dry neck. You need a succulent neck. So whatever we do to reduce the dryness of the neck will impede bacterial ingress from the foliage to the bulb. Okay. When you grow onions have deeper roots. How do we harvest? We pull them. The pulling is one method. Another method is digging. So what you do, you use a chain digger that digs and uproots onion, or you can use a underblade undercutter. Means you have a blade that goes inside the soil and cuts. What we have seen, when we use an underblade undercutter, you'll have a more internal rot compared to chain digger. Why? Because chain digger uproots the roots. When the roots are uprooted, do you think the neck of the onion will be succulent? No, because it has no water contact. It cannot uproot, cannot take water from the soil. So if the neck is dry, do you think the bacteria will move from the neck to the bulb? Not at all. So that's the reasoning here. Whereas when you do the chain digging, it's uproots, resulting in complete removal of the roots from the soil, so that a neck dries really fast in that process. As a result, you have a less internal rot. These are very small changes you can do. All right, last slide. So something harvesting, the way you harvest, what we have shown, if you do a mechanical harvesting, you use a mechanical harvester to harvest onions. It cuts the onions at a typical neck length. When you cut the onion at the typical neck length, at least 2 inches, and it's dry, it will reduce the neck rot. Whereas when you tell your workers to cut onions, they will cut at varying length keeping varying neck length. If they cut very close to the onion shoulder, you'll have more internal rot compared to others. So, well, that's what I have. If you have to leave at least 2 inches of neck length to have a less internal rot. Now, this information is very important for Indian onion growers too. This has been This has been disseminated in the United States, and it has been widely accepted. So this is the way small cultural changes can reduce bacterial internal rot, and as a result, can have more revenues. With this, I'll go to my last slide, if I can move. This is my group. This is my 5 postdocs, 5 graduate students, my collaborator, the old man over here. He is the world-renowned bacteriologist. He is my collaborator, and my another collaborator, Dr. Brian Koitko from University of Georgia. My funding agency is my whole group. And with this, I'd like to take any questions if you have. How long did I take? Well, red. Very good question. Let me answer you this thing. Let me answer the second question first. Pentoea is ubiquitous. It is present. It is an environmental. There are strains which are present in the environment. There are strains which can cause disease in animals. There are strains that cause disease in humans. There are strains that cause disease in plants, and there are some also biocontrol strains. But there is a clear demarcation between these strains. Seldom we see overlaps. Okay, but there are some strains. I'm going to tell you the story that if those phosphonate cluster, which is present in the plasmid, somehow it can get horizontally transferred to the human strain, that particular human strain can also become plant pathogenic strain, and vice versa. Yes, so it is very rare. I have never seen. I have the largest collection of Pentoea in the world. So I collect onion bacterial consortium in my lab. None of them overlaps except few, which we have to be very, very careful.
Biosafety.
Oh yes, so
You
I'm
cannot
not
recommend Pentoea for biocontrol.
very good question. Pentoea biocontrols a little. I would use it if it's labeled by the company, because there's a lot of multi-location trials, a lot of years of trials have been done. And again, I'm going back to my previous assessment that we seldom see overlaps or crossovers. Very, very rare. Now, what was your first question? What was regarding red onions? Okay, you have a yellow onion, you have a white onion, red onions, different. Which onions will have more sulfur? Which make you more dry? Red onions. Red onions has high sulfur contents. So those Pentoea that contains all the genes of allergen tolerance will be much more suited there. So red onions are little tolerant to Pentoea, but we do have strains that can rot red onions like anything. Most of the work was done over there on red onions. You see all the red onion slides. We did not use the yellow onion or white onion. So anyway, the long story short, some of the information we generated can be disseminated here for the onion growers in India too. So any more questions for me?
Okay, thank you. We shall discuss during dinner time.
Oh, sure, thank you.
So sorry, we could not give you more time. So thank you.
Thank you all. Thank you for listening and the patience.
Next.
Thank you, sir. Next presenter is Dr. Apana Patekar, Juniper Cake Tipton, Georgia, United States. She's not there? Okay.
It's not there.
Yeah, oral presentation. Okay.
Next, Adesh Kumar,
Dr. Rakesh Kumar Singh. Oral presentation. Number 1,
No, no.
Dr. Rakesh Kumar Singh.
There, Dr. Rakesh Kumar Singh. Acha, okay.
Okay, thank you. Respected chairman, co-chairman, devotees.
Try to crisply, if you can present,
yeah,
no,
within-
to help all the other
Of course,
speakers
sir, within 10
and
minutes.
organizers, please.
This is my oral presentations on investigation on progressive systemic localization, seed-borne nature, and symptom-based advancement of Fusarium oxysporum form species Siceri,
That one pathogen is known for causing disease in chickpea. And basically this pathogen is soil-borne nature. But sometime many reports are there. The pathogens reached up to seeds. So how the pathogen reached up to seed? So this all the studies only concentrated on that matter. So wilting in chickpeas caused by the combinations of pathogen activities such as toxin production, accumulation of fungal mycelium in the xylem vessels, productions of gums, gels and tylosis, and vessels crushing the by proliferating adjacent parenchymatous cells. And under field condition, two types of wilting symptoms generally observed. It's a early wilting and late wilting condition. And these symptoms are usually more visible in the early wilting symptom visible in 6 to 8 weeks after sowing, and late wilting symptoms appear after that. And systemic progression of pathogen in different genotypes was evaluated based on the occurrence of infection at 5 different stages of pod formations, such as infection at less than 10%, less than 25%, 50%, 75%, and 100% pod formation stage. And detection of Fusarium oxysporum caesari from 31 late wilter plant seeds were carried out during the studies. And this is the late early wilting conditions generally observed under the field condition. These all are the wilt seed plot appearance of the disease. And here in the second right hand picture, you can see the late wilting lines which express the wilting at the late seasons. And these are the some genotypes which showed the 2 types of symptoms, that is chlorosis and drooping type of symptoms. So these are the lines selected for the studies. And these are the lines selected for further systemic progressions of the pathogens studies at less than 10% pod formation, 25% pod formation, and 50% pod formations. And some resistant genotypes we have also taken for comparisons. And this is the critical drooping symptoms generally observed under the field conditions by the chickpea. That is the drooping start from the apical portions of the plant. But in other types of symptoms, wilting and chlorosis start from lower parts. So that is chlorosis symptoms initiated from lower parts. So this is the major differentiation in between the 2 types of symptoms produced by the chickpea wilt pathogens under the field condition, or you can say under the wilt seed conditions. And this is the differentiations in xylem vessels discolorations. Here you can observe that the different types of discolorations patterns observed during the in the sowed drooping symptoms and chlorosis symptoms. Here we collected some of the seeds of the different around 31 late wilter lines. And the next year we detect the seed-borne pathogens from the seeds. And this is the recovery of the Fusarium oxysporum from the different genotypes we have which collected in the previous year. And we generally detect by using the 3 methodologies, that is generally agar plate method, standard blotter method, and rolled paper towel methods. And all the 3 techniques of detection, seed-borne detection techniques are equally effective for detection of seed-borne natures of the pathogens. But rolled paper towel method are more efficient to detect the seed-borne natures of the pathogens from the infected seed. And this is the associations of the seed from the different plant parts. And this one pathogen is not externally adhered with the seed surface, but it's associated in the embryo and cotyledon. And this is the isolations from different parts of the seeds and the different parts of the different parts of the plants. If you observe, we isolate the pathogens from the lower parts, middle parts, and seed. So these pathogens in the systemic way cause infection and reached up to seed through the systemic way. So this study is clearly revealed that the systemic infections of in late winter conditions, pathogens reached up to seed. So that is the reason why this soil-borne pathogen become seed-borne. Here, if you see the showing mean systemic infection in root, stem, branch, and seed portions of chlorosis and drooping side symptoms, so there are very minor differentiation in between the two types of symptoms generally observed under the field conditions. If you see the recovery of the pathogens from the roots, so in both the symptoms, it's around 90% recovery. In stem, it's a 60-70% recovery. But if you see the recovery from the seeds, so that is drastic differentiations are there on the basis of both types of symptoms generally observed under in field conditions. So it's a chlorosis types where the chlorosis type visual symptoms observed under in field conditions, where more recovery of the seed-borne pathogens are reported as compared to drooping type. Means if the plant showed chlorosis type symptoms, that means there are a strong possibility to transmit the seed pathogen up to seed as compared to drooping plants. Here we actually, it's a systemic progressions of the disease in root, stem, branch, and stem respectively at 5 different stages of pod formations. It's a root, branch, stem, and seed. Here if the infection appears at the time of 10% of pod formation, so that will prolong up to 36% recovery of the pathogen. And if the infections occur in 25% pod formations, that around 33% recovery of pathogens. And if the 50% pod formation infection occurred at the time of 50% pod formation, so that will lead to 12.5. That means if the infections occur at later stage, so that will drastically reduce the colonizations of the pathogens up to seed level. Here, chlorosis recovery of Fusarium oxysporum from different parts of chlorosis and drooping symptoms plant. Both the symptoms have the equal level of recovery from the collar region, first branch, second branch, third branch. The different parts of the plants we use for isolations of the pathogens for see the colonization pattern of the pathogens on the basis of both type of symptoms, that is chlorosis or drooping type of symptoms. Here you can observe the both symptoms, drooping type symptom, drooping type symptom generally start from the apical parts of the plant, and the chlorosis symptom start from the lower part of the, generally chlorosis or necrosis symptoms appear in the lower parts of the leaf. So that is the major differentiation in between the two types of symptoms. And for conclusions, the early and late wilting symptom expressed by the chickpea plant under natural epiphytotic conditions depends upon the available host resistance and inoculum load of the Fusarium oxysporum caesarea. In the late wilting condition, two types of symptom expressed by the plant, that is drooping type and chlorosis type. And these symptoms initiated from lower leaves and vice versa. And systemic infection or progressions of pathogens to different parts of plants was highest in plants showing chlorosis symptom, that is 32.5%, when compared to plants showing drooping symptom, that is 7.5%. So there are the differentiation on the basis of visual symptoms appear under the field conditions for the colonization or for advancement of the seed-borne seed pathogen to the seeds. And different late winter lines were collected for study of progression infection in parts like root, stem, branches, and mean systemic infections in the roots is the 93%, stem 74%, branches 44%, and seed 32.5% recovery of the pathogens from different plants of the parts. And infections in the seed was highest when the plant were infected at 10% pod formations. So if the plant get the infection at the early stage, at the time of 10%, that will lead to more seed-borne, nature of the pathogens, and was lowest when the plants were infected at 100% pot formations, because there is very less time and by which the pathogens cannot reach up to seed systematically. The pathogens survive internally in the seed harvested from the late wilter plants and vice versa. And mostly early wilting occurs in the field initiated on plant by the primary infections of pathogen through seeds, but depends on the host resistance, quantity of viable inoculum, load present in the seeds, and epiphytotic conditions of the soil. Among the three methods of detection, agar plate method, blotter, and towel method, rolled paper towel method was comparatively best one. That's all, sir. Thank you very much, sir.
Thank you, Doctor, for excellent work and excellent presentation. Thank you.
Thank you. Thank you very much.
So now we call upon Dr. Sajadun Nabi. So Dr. Adesh Kumar is there. He's not there. I think Adesh is not there.
We'll call up our next speaker, Dr. Sajad Ud Nabi, ICR, Central Institute of Temperate Horticulture, Srinagar, Jammu and Kashmir, India.
Respected chair, co-chair, and other participants, I welcome you all in my presentation, which I'm going to be delivering on real-time detection and transmission of viruses associated with mosaic disease of apple. So if we see, apple is a very important and remunerative crop which is grown in the temperate regions of our country, and it is a mainstay of economy for the farmers, and we are generating almost 15,000 crores from this apple cultivation. So if we see the virus spectrum associated with this crop, almost 19 viruses have been reported and 5 viruses have been reported till date, which are affecting this crop. And there is a clear cut that the virus and virus-like diseases reduce the yield and productivity of this crop up to 25%. So among the diseases, apple mosaic disease is a very important disease, and it can reduce yields up to 30%, and the photosynthetic activity it reduces up to 50%. When photosynthetic activity is reduced, then obviously it will reduce the yields of the crop. And if we see, the causal agent of this disease was apple mosaic virus, and it is an ELAR virus. Earlier it is apple mosaic virus, but now the scenario has changed, which I will discuss in future slides. So there were few reports from China and from Korea. There were few reports of this prunus necrotic ring spot association with this mosaic disease. But from China and Korea, there was a new virus, that is apple necrotic mosaic virus. That was a new virus associated with this disease. So far as in Indian conditions, we don't have the information whether this is apple mosaic virus or apple necrotic mosaic virus. So we tried an approach that was the next generation sequencing approach. And we selected 3 cultivars of the apple. One was asymptomatic, which was not showing any kind of symptom. And then 2 symptomatic cultivars, one was the Oregon and another was the one was the Golden Delicious. You can see here in Oregon Super, there is necrosis as well as mosaic, but in Golden Delicious, there was only mosaic type of the symptoms. So after applying this RNA sequencing approach, what we found that in red Fuji, which was asymptomatic, the apple mosaic virus or apple necrotic mosaic virus was not present. But to the best of this Oregon Super and Golden Delicious, which were having the symptoms of mosaic or mosaic necrosis, you can see there was apple necrotic mosaic virus, which was earlier found in the China and Japan or Korea, but we found in our cultivars as well. So in order to validate these results, what we have found through NGS approach, we surveyed various districts of the Kashmir Valley where apple is grown. And what we found that after going for the ELISA for apple mosaic virus, that is DAS ELISA, what we found out of almost 20 or 30 samples, only 2 samples we could found positive. But through RT-PCR, again, when we go for the apple mosaic virus, out of 20 samples, 2 samples were again found positive for this apple mosaic virus. For What about the apple necrotic mosaic virus? Out of those samples, when we amplified this virus in those samples, we got amplification from almost 50% of the samples infected with this apple necrotic mosaic virus. So from this survey and RT-PCR results, what we could infer that this apple necrotic mosaic virus was mostly this abundant virus present in our Indian conditions. So then we know that these viruses belong to the LR virus group, and we can say that is isometric labile and rings labile viruses. So their titer varies with the temperature, and their distribution in the plant is also varies from one part to another part. So we designed the study that how we could to study to spatial and temporal distribution of the viruses, both the viruses, apple mosaic virus and apple necrotic mosaic virus, so that we can choose this tissue as well as season, because we have 3 distinct seasons in our temperate regions, spring, summer, autumn, and winter. So which will be the best season, which will be the best tissue, so that we can use those tissues and that season where we can have the indexing program of these viruses. So these were the tissues which we selected for indexing or for this detection through RT-PCR and real-time PCR. These were the tissues: leaf, flower, bark, fruit, seed, as well as the pedicel of the leaf. So in different seasons we detected. The spring season was the best season where we could detect in all the parts. What we have targeted in all the parts, we could detect both the viruses, apple mosaic virus and apple necrotic mosaic virus. But in summer season, we could detect only in the leaves. We couldn't detect in this pedicel, we couldn't detect it in leaves, but only in the seeds, both the viruses. And then in the autumn season, again, we could detect these viruses again in the leaf, because these LR virus, there is a negative correlation with temperature, and we could observe through RT-PCR as well. Only we could detect the viruses in seed, which embedded inside the fruit, but couldn't detect in the these other parts like leaf and pedicel in the summer season. So to quantify, we did the relative quantification through real-time PCR to know the titer of the virus, which is present in different tissue parts, in the different tissues of the plant. So we did real-time analysis as well, and we could found where we could not found the virus through RT-PCR in summer season. We could detect the virus through real-time PCR in those tissues through real-time PCR. So from this study, the titer of the apple mosaic virus and apple necrotic mosaic virus more in leaves, followed by flowers in spring season, wire in seeds in summer season, and leaves in autumn season. And also the leaves and flower during spring season, seeds during summer, and leaves in autumn season can be used as a detection material for early and rapid detection of both these viruses. Then we could, because this was a new virus to our country, so we could design this experiment how it will get transmitted. Because this apple is a colonially propagated crop, and the virus also gets transmitted through signwood or budwood. And we conducted one experiment through, we tried to transmit these viruses through this grafting and budding. So we selected 8 rootstocks, and on different rootstocks of apple, we have different rootstocks of apple. We selected 8 rootstocks, and on which we grafted the virus infected this signwood from the virus infected plant. And what we could observe, as well as budding we could do on those rootstocks, what we could observe that the grafting transmitted both the viruses. Budding, the transmission efficiency was less as compared to the grafting experiment. So grafting can through this grafting approach we can transmit both the viruses, as well as through budding approach we can transmit the viruses, but budding has a less efficiency than grafting, what we have observed in our experiment also. So these are the conclusions from my study, that the RNA sequencing assay detected a new virus, that is apple necrotic mosaic virus associated with mosaic disease. Then the PCR-based results not only validated the high throughput sequencing results, but also showed association of this necrotic mosaic virus was more abundant than apple mosaic virus. And leaves and flower during spring season, seeds during summer, and leaves in autumn season can be used as a detection tissues for their detection for both the viruses. And viral load through real-time quantification will help in selection of tissues for virus indexing of both the viruses for certification program. And the comparative transmission efficiency was highest in grafted plants than budded plants. And both viruses were confirmed from these grafted and budded plants through conventional RT-PCR with both the viruses. Thank you very much.
Thank you. Thank you. Thank you.
Thank you, sir.
Thank you for presenting in time. Next presentation is by Dr. Sajina from Kerala Agriculture University. So, please try to complete in 8 minutes.
Okay. Yeah, this is forward and this is forward.
Respected Chair, Co-Chair, dignitaries, and my dear friends, I am Dr. Sajina from Kerala Agriculture University, and I welcome you all to my presentation on new report of a bacterial wilt disease in the bush cowpea caused by Klebsiella pneumoniae. We know the importance of bush cowpea. It's an important leguminous crop, and it is grown for both seeds as well as vegetable. And it has importance as it has proteins, vitamins, etc. And it is also used as a food, feed, fodder, and green manure crop. It also has economic importance because it is resistant to drought, it conserves moisture, and it also prevents soil erosion and weed growth. So in our station, we have a pulse hub program in which different types of pulses we grown. And in the bush cowpea, we could see different types of diseases. For example, the most important were the basal stem rot caused by Sclerotium rowlesi, an attack of shoot and fruit borer, and fruit rot caused by Coenophora, and the pulse beetle. But among the field, there was a wilt disease of the bush cowpea plants, especially the variety Kanakamani. And there was 60 to 70% incidence of the disease. And the symptoms were drooping of the leaves, collapse of the stem, and the death of the plants. And water streaming test or the ooze test is confirmed the presence of bacteria. And so there was a necessity to identify the pathogen. And so the objective of the work was to identify and characterize the bacteria. Standard procedure was followed for isolation of the pathogen. So this is the pathogen which we isolated in TZC media, and single colonies were streaked to get pure culture in CPG as well as TZC medium. And pathogenicity tests were done by two methods: root dipping method, in which the seedling was immersed in bacteria grown culture, and soil inoculation method, in which the bacteria was grown in CPG broth. It was kept in the rotary shaker and it was incubated for 1 week, and the plants were grown in the bacteria inoculated soil, and within 5 to 6 days there was the symptom expression. And we proved the Koch postulate by streaking the pathogen, and this is the recovered bacteria which is similar to that of the original bacteria. Next, we wanted to characterize the bacteria. On gram staining, it was found to be a gram-negative bacteria with a rod shape, and it was positive to KOH as well as catalase test, which proved it to be a gram-negative bacteria. Now we wanted to go for the molecular characterization. We were already working with Ralstonia. So we thought the bacteria to be Ralstonia. So we did with specific primer of Ralstonia, but we didn't get the result. So we did 16S RNA and we got it as Klebsiella. Specific primer of Enterobacteriaceae was taken, and among that, RNA polymerase beta subunit, RPOB gene was taken, and the locus was amplified, and we got 100% similarity with different isolates of Klebsiella, and the gene sequence was deposited in the gen bank. So this is the blast with RPOB gene. We got 100% similarity with Klebsiella pneumonia. We did phylogenetic tree with sequences of the RPOB gene, and our contic, that is the bacteria of interest, clustered with Klebsiella pneumoniae. And this is the barcode gene gaps, which showed that our bacteria was found to be exactly similar to that of Klebsiella pneumoniae. And further, we also did biochemical tests. There is a kit which is specific to members of Enterobacteriaceae. There are 24 tests. The first 12 tests are for biochemical, and the next 12 tests are for carbohydrate utilization. So to differentiate between the Klebsiella and the Ralstonia, there are 5 important tests. First is esculin hydrolysis. Klebsiella was positive, and Klebsiella could ferment the glucose to acetoin. Arabinose was positive, but it doesn't have a cytochrome oxidase, so it is negative, and it produces hydrogen sulfide. So I'm just showing a paper published in 2022 in China. It shows that Klebsiella pneumonia not only attacks human and animals, it's also a plant pathogen. Infection in maize, banana, and pomegranate has also already been reported. And there is a gene called RCSB, which is actually responsible for the kingdom crossing. So these are the reported diseases by Klebsiella all over the world. This is the report of the diseases by different members of this, especially Cosaconia. And in the study, I conclude that bacterial wilt has been observed as an important disease of bush coffee with 60 to 70% crop loss. It's a rod-shaped, gram-negative, and facultatively anaerobic bacteria. And the bacteria was identified to be Klebsiella pneumonia. And this is the first report of this pathogen in bush cowpea. And I just want to share another more work which was undertaken in our lab. This is a bacterial wilt in the same type of cowpea, which is a trailing type. This type of wilt was observed, and we thought it again to be Ralstonia. These are the various symptoms, and the pathogen was isolated. But with the 16S RNA, we could get it as Enterobacter. So we did for the specific RPOB gene again. The bacteria was found to be Cosaconia. Some of the species of Enterobacter has been renamed as Cosaconia. So we got it as Cosaconia oryzae, and the phylogenetic tree also shared it with it. And these are the various pathogenicity tests we undertook. And this is also a first report of the bacterial wilt caused by Cosaconia oryzae. And I want to just share that we have to be very careful because bacterial wilt, the pathogen has been understood as Ralstonia, but several papers have been published which shows that the most important bacterial pathogen is Enterobacter, followed by Klebsiella, Pantoia, and only finally is the Ralstonia. And I extend my gratitude to Kerala Agriculture University and IPS for giving me an opportunity to present. Thank you.
Thank you. Thank you for good work and good presentation. Thank you.
Wait. Sajan. See, you have worked with the pathogens of both the hosts, which are human pathogens.
Yes.
And how careful while you are handling these bacteria?
We were careful saying we were thinking that it was Ralstonia, sir. And later onwards, we were very careful with this because we know it was causing human pathogens.
Did you test in the soil or your vegetable crop block where these cowpeas are grown? Did you check in soil presence of these bacteria, not in the host, in the soil?
No, sir. We did from the host because it took so many years, 1.5 years for the identification, so because it was so confusing. So from the host plant only we isolated.
Not in the soil. But the second person can continue with a care,
Okay.
not to get infected.
Next one, Dr. Vijayanti Mala. Vijayanti Mala from Mauritius. No? Dr. Sudha from Central Coffee Research Institute.
Sir, 1 minute. Is Dr. Aparna Patekar is present? No. Dr. Adish Kumar, Lovely Professional University. Is he present? No. Okay. Then Dr. Vijayanti is also there. Okay. Then Dr. Sudayam. Yes. Over to you, ma'am. And one more. This one announcement to be made. There will be a gala dinner today evening at 7 at Hotel Lair Ruchidi Prince, and a pickup and drop will be done by the same venue. I request all the participants to join for the gala dinner.
Respected chair and co-chair and rapporteurs and dear delegates. First of all, I would like to thank the organizers for giving me an opportunity to present on the topic diagnosis of emerging fungal pathogen and Robusta coffee. So as in India, nearly 90% of coffee area lies in southwest monsoon dominated areas. Remaining is mainly at higher elevations of northeast monsoon influenced areas. Coffee cultivation in India is unique as it is grown under multi-tier shade in a simulated microclimate. And moreover, coffee plantations are enriched, not only the coffee, with suitable intercrops like black pepper, areca, orange, avocado, banana, etc. And as in the coffee, there are many, important diseases. As we know, coffee leaf rust is one of the major important disease. Apart from that, during monsoon season, we encounter, a few monsoon rots. Normally, as I said in the previous slide, the southwest monsoon, that is, invariably during the southwest monsoon season, the coffee plantations, receives heavy downpour from June to August. So during that season, nowadays, what's happening is we are receiving unusual rains, maybe because of the climate change. And what happens to the unusual rains? So that is staggered flush development is noticed, and multiple blossom is noticed, and existing pathogens are activated. That conditions are ideal conditions for flare-up of the disease. And normally during this monsoon season, the coffee plantations invariably suffer from the fungal diseases like black rot and stalk rot of diseases. Black rot, this is the first, the other one, the left side of the PPT, that's black rot, which is caused by Chlorogonoxia, and the other one is stalk rot of coffee caused by Coleotrichum gliosporides. And we can see the rainfall, as I said, due to the climate change, we are seeing, there is a change in the rainfall distribution as well as the pattern of rainfall what we receive. We can see here we have taken some more than half a decade or 5 years of data, 5 to 6 years of data. You can see from June to September, we are receiving high and erratic rainfall. We can see during August, during 2019, we can see 1,204 mm of rainfall is received within a span of 31 days, wherein what happens to that? That is heavy rainfall within a very short period. So what happens is we can see the stagnation of water when heavy downpour is received. We can see the coffee plantations, immersed. The plantations are immersed in the water, and we can see afterwards, maybe 1 or 2 days, if it is, the drained out water, then we can see the effect of monsoon season. You can see the yield loss, even defoliation, as well as, premature leaf fall, as well as premature fruit drop is seen. And in addition to this is a preamble. In addition to these two diseases, we are noticing one more disease. Disease is emerging in this last 2 years. So this is the emergence of new disease in addition to the black rot disease as well as the stalk rot disease of coffee. And here we could able to see, the planters were requesting us to, say that during monsoon season, there is plenty of mealybugs. That's why report we received. Then after once the monsoon is, cessed, we went to see the estate, and naturally we could able to see that there is fluffy and white mycelium growth is there, and then the stalk, even the fruits are infected. We can see abundant white mycelium on the infected parts of almost all the leaves, even the fruits, as well as the young branches. And then what we could see is the characteristic symptoms of the rotting of berries with prominent white mycelia and brown color sclerotia on berries and all along the twigs. And these disease samples were collected and pure cultures from these were obtained. And normally this disease somewhere during early 80s, this was been recorded as soft rot of coffee in Arabica plantations. But afterwards, we never have noticed, even I have joined somewhere in 1993 to the coffee board, but I never have seen such a condition, the specimen itself. But this is from 2 years we are observing in the estates because of the inundant and unprecedented rainfall and climate change. So this is a first record on Robusta coffee. And it has been cultured and general media, that's a potato dextrose agar medium. And we could see the white color, fluffy and fibrous mycelial growth. And sclerotia were, just like, mustard like sclerosias were observed. And these sclerotial in a culture plate, it was restricted to periphery and most in the center and periphery. And yes, this. And then to prove the course postulates, pathogenicity test was carried out, and we could see in the glass house we have inoculated the pathogen, and then we could able to isolate from the leaf the typical symptom of that, and then we could able to re-isolate the pathogen. And what is the concern and what is the issue of that? Though it looks very simple, only in the monsoon season we are observing this disease. What is the concern of this? Really, in addition to the monsoon diseases, now almost the coffee planters from in South India, from 3 to 4 years, we are claiming for the subsidy for premature leaf as well as fruit drop from the government. It is a great problem, and always we. As a pathologist and as a group, we used to go for assessment of all these diseases. And again, this is really a major concern as it is emerging, and now the climate change is also occurring. And again, the survival of the pathogen, as we know, this is in the form of sclerotia. This pathogen survives for many years. Even in the introduction of this plenary session, one of our keynote speaker told it will be there for 10 years. So here it is not in the soil, but it is in the host plant. So several years it will be, survival of the pathogen will be there for several years. Even then, environmental conditions, of course, for this pathogen, as the climate, it favors, of course, now we have some high temperature, naturally followed by humid conditions, and the soils and the acidic soils is there. So that is a favorable condition for the flare-up of this pathogen. And due to all these things, definitely there is a yield loss. We have seen the entire branches, entire bunches, entire clusters of the coffee has been dried because of the infection of these things. And definitely due to that, deterioration of the quality is seen. Since it is exportable commodity, it is very important to note the quality of the coffee. Again, the residues, whatever it is left on the plant, definitely it affects to the next year crop. That's a major concern now, in addition to the 2 diseases. And in future work, it has to be in line with, we need to take up the complete epidemiology of the disease, and we need to see the climate resilient techniques to combat the disease. And of course, management we have taken up in the pilot studies, and we have recommended the planter to go for 1% border mixture. But when, in February and March, we'll be receiving some rains, blossom showers, so that the pathogen which is there on the plant, it will become active after receiving the rains. So before to the onset of monsoon, we have recommended to take up 1% border mixture, but still we need to take up a detailed study on the management, since we always restrict our research to the need-based problems rather than the academic ones. And then finally, we may go for molecular identification. Thank you.
Thank you. I think that completes the session. So we have successfully completed the session, which had 3 keynote lectures, 3 invited lectures, and 5 oral papers. And of course, enormous information has been generated on that for the audience. And I thank our co-chair and also the rapporteurs for ably supporting in the conduct of the session. I leave the mic to our co-chair.
Thank you, Chairman. It was a fruitful session. We could able to manage within 3 hours. But only I feel sad the presence of the even students are not there. All see, we are senior most professors who are there, sitting 3 hours. The youngsters, I see very few people. I think this information youngsters will not get anywhere in the books or journals. But these conferences are meant for their knowledge enrichment, and I find most of the youngsters are not aware and they are not present. This should change. And I thank the organizers and chairman for giving me this opportunity. And with this, we close the session. Thank you, one and all.
Thank you, sir. At this, we came to the end of the session. And I request Dr. Chahal, sir, former President IPS, to present mementos to our chair and co-chair and rapporteurs. Chahal, sir. Thanks. Chair, Dr. R. Vishwanathan, ICAR, ISSR, IISR, Lucknow, Uttar Pradesh. Yeah, please join. And co-chair, Dr. M. Krishna Reddy, ICAR, IIHR, Bengaluru, Karnataka. And rapporteur, and the rapporteur, Dr. Prakash TL, ICAR, IARI, Indore, Madhya Pradesh. Thank you, sir. Thank you all for your patience, hearing, and presence. Thank you, sir. Chahal, sir. Thank you, sir, for presenting mementos. And once again, I remind there will be a gala dinner today night at 7. Pick up and drop will be available for all the participants. Thank you, sir.